find the information after a certain perturbation or change into the cell. And therefore, metabolomics is very useful to investigate the cause of disease. What happens, and it might change the metabolome of a particular bioproduct after attack of a disease. And then we could come up with ideas on what is the cause of the disease and how it could modulate the metabolome, and then how we could inhibit the spread of the disease. And we could also use this to assist the development of post-harvest management. And then afterwards, we could also help to improve the quality. And these are the points that I will be discussing today on how we could apply metabolomics, particularly to solve the problem of crop loss and also to improve the food quality of Indonesian agricultural products. So the concept of food metabolomics is very, very simple. It's basically correlation between a specific quantitative phenotype that could explain taste, grade, palatability, or whatever as a quantitative phenotype of a particular food or agricultural product. And then we could correlate that with the analysis of metabolomics, where we could quantitatively measure the concentration of the components in a comprehensive manner. And then afterwards, we have many information of the metabolome of a particular sample. And then we have the information of the quantitative phenotype. For example, this particular sample has a high bitterness or low acidity or something like that. Then afterwards, we could make a correlation using a regression model. And we could explain what makes a particular food taste a certain way. We can then explain what is responsible metabolite that is responsible for food quality. And then afterwards, we could then further investigate what makes the concentration of this metabolite, which is responsible to food quality, high in a particular sample, and how we could engineer the process in order for us to improve the concentration of this metabolite, which will later on correspond to quality. So as a result, through my research, about 10 years of research experience dealing with metabolomics, I have already expanded my research collaboration, particularly in the medical and pharmaceutical science. To the latest one is the collaboration with Harvard Medical School to combine the information that we have from Indonesian bioproducts and the effect of these bioproducts, the hidden health benefits of our bioproducts. And with the Harvard Medical School, they did a lot of studies, also with cohort studies, with human intervention, diet intervention, and then we could find new health benefits in our Indonesian bioproducts. And I also dealt with microbial strain improvement, in particular for the production of different biochemicals, biofuels, very high commoditive, high economic value commodities in collaboration with RWTH Aachen, with NUS and UCLA. And for the food and agriculture, I focus only on Indonesia and collaborate with a lot of partners. And most importantly, we also collaborate with BRIN, ORPP, which is the host of today's international conference. So as I have already mentioned, Indonesia has a very rich source of bioproducts, food products, and therefore I came up with ideas to analyze different agricultural products, aquaculture products, and fermented products. I think this is a representative from marine, from sea, as well as the involvement of microbe. This represents the various kinds of biomaterials and food materials that we have in our country. And therefore, we would like to apply metabolomics technology for many different kinds of research area, including to improve the shelf life of food. And this is very important for us to prevent food loss, how to keep food last longer. We also improve the quality. We establish the method to do authentication of a bioproduct. We do diet monitoring, and also diet monitoring in terms of investigation the effect of a particular diet in human metabolism. We also identify unique characteristic of our products and do the product development. I think these last 2 points is very, very important because Indonesia urgently needs information, detailed information, quantitative information, scientific information on what are the unique characteristics of our byproducts. With our very, very diverse bioproducts available, we need to be able to create a good database so that we would know what is the important unique characteristic of a particular coffee, for example, coming from a certain region, and how we could use that kind of information for us to promote our byproducts further and do the product development. So I would like to focus on these issues related to SDGs as well. One of the key points of SDG is to achieve food security and improve nutrition. So improving the quality, promoting sustainable agriculture, achieve food security is one of the most important goals of SDGs. And of course, also food is a very important component for health and well-being. And therefore, healthy food, healthy diet is also very important to promote well-being, which relates to another point of SDG. And finally, at the end, to realize reasonable consumption. So this is also to minimize food waste and to optimize the production process of food. So here I would like to talk about the food loss reduction by metabolomics approach. So as have been already known, perhaps by many people, actually we have a very serious issue of food loss. If you do not know, I would like to tell you that actually one third is 33% of the world's food production is wasted, and the economic loss due to that accounts for $7,000 billion per year. And actually, if we consider how much food there is to throw away and without being eaten, is actually enough to feed 2 billion people in the world. And of course, it also caused a major problem about carbon dioxide emission, which result in global warming. And also, therefore, we would like to focus on the solving this problem of food loss by having a target using our project to apply metabolomics and also other AI technologies. We would like to reduce the food loss along the production supply chain, including post-harvest losses. So we would like to focus on certain points that is related to food loss. Here we could divide the food loss into production loss, storing loss, transportation loss, and etc. And we would like to optimize each of the production process in order for us to reduce the food loss from each of the different category. And we would like to do that in collaboration and support with the Indonesian government, particularly with BRIN. Why we would like to focus on Indonesia is it's a very sad thing. Actually, now Indonesia is second largest per capita food waste in the world. We produce so much food, but we waste so much food. But also at the same time, there's an opportunity because we are also the largest fruit producing country in Southeast Asia. As you may know, we have a very high fruit production, but fruit is highly perishable material, and therefore focusing on optimization of the post-harvest strategies in fruit will enable us to minimize the risk of food loss. And hopefully, fruits for Indonesian only, if we could reduce the food loss and waste for worldwide, it can account for 0.3%, which approximately accounts for $22 billion. As a result, I have already started research starting from a very good tropical fruit unique to Southeast Asian country, mangosteen. I work also with coconut, different cultivars of coconut, different cultivars, different regions of pineapple. And we collaborate also with Indonesia for banana. And lastly, we collaborate also on mango, which is a result of our collaboration with RPP BRIN here under Dr. Puji Lestari. So here, we would like to focus also in this project to monitor what happens during the process from harvesting to consumption. So this is the banana supply chain. And one of the points that we would like to focus to develop in this project is to have a very smart tool to monitor the changes that happens and minimize the risk of food loss. The main idea is that during the marine transportation, we would like to have a good tool to monitor the changes that happens and mark the onset of spoilage, early ripening, and etc., or infection, so that from the farm to aggregation point, before doing the marine transportation, we could prevent the loss before shipment, and then we could prevent a bigger loss later on during storage and distribution if we have already identified early markers of spoilage before transportation. So one of the points that we would like to develop is to monitor like kind of marker and tool. And we would like to have a very nice low-cost tool to monitor the quality of fruits in the field and during the transportation and just before the consumption. So to do that, we also collaborate with key stakeholders. This is a collaboration with ITB. We compare different types of post-harvest treatment to compare how we could prevent the ripening of banana, delay the ripening of banana. Here we employ low temperature treatment, which is a very useful technology to prevent ripening by low temperature treatment. This is a very common one. And also, but at the same time, this low temperature treatment requires us to have electricity and very expensive cost for instruments to keep this running. And as you may know, in Indonesia, all over Indonesia, the availability of electricity, stability of electricity current is not so stable. So the ITB team, Dr. Feni, has already developed a method using chitosan treatment to delay the ripening in banana. And then both of them shows the delay in ripening, but using metabolomics, we could understand further the mechanism by which the delay happens. And it turns out it happens using a different mechanism. We found that when we compare the low temperature treatment and the chitosan treatment, the different mechanism occurred. And for example, different kind of metabolites were affected by low temperature treatment compared to the chitosan treatment. So the chitosan treatment affects the metabolite related to ethylene biosynthesis. By inhibiting the ethylene biosynthesis, the ethylene production is also decreased, and then this also delayed the ripening. But the low temperature treatment affects the metabolome in different way, which is related to hypoxia, a low oxygen condition under this treatment. And we would like to have an idea also on identifying this particular problem in Indonesia, because we have so many different kinds of cultivars of our fruits. For example, when I started this research, I would like to ask whether there's a quantitative way to determine the differences between our most famous mango cultivars. And it turns out I couldn't find any literature that describe what is scientifically and quantitatively we could describe objectively the difference between Lalijiwo, for example, with Arumanis or Gedong Gincu. So I thought that it's probably useful for us to monitor or to measure the metabolome and then compare the metabolite concentration of these 5 different cultivars, and then we could find the unique points of each one. Then once we collect also the information, we found that mango that is Arumanis and Gedong, actually, in fact, Podang have high sensory score, whereas in this study, we found that the other 2 cultivars have lower sensory score, but Indonesia produces a lot of these cultivars. Then how we could make use of these cultivars once we know what is the characteristic of the cultivars that is less preferred and what are the metabolites that are responsible for the off taste or the taste that is not preferred, we could change or modulate this metabolite in mango and process it so that we could minimize the concentration of particular organic acid that makes it too sour. And then afterwards, we could make this mango into different product so that we could minimize the risk of food loss. So we are now doing the research to study the metabolite changes after the processing, and we would like to suggest that as a strategic way for us to optimize a way for production process of these mangoes that are high in production, but are less preferred and have a very high risk of food loss. So next, I would like to describe, after the story of food loss, I would like to describe on how we could use metabolomics to improve the quality of food. And I would like to use this opportunity to share my story about the research on different kinds of studies in coffee. I have already comprehensively analyzed on how to do the authentication of kopi luwak, what kind of metabolite is responsible for coffee quality, what are the factors that are important to modulate metabolite concentration by analyzing many different kinds of coffee species coming from different origin, coming from different altitude that were processed in different post-harvest processing, for us to get a very big picture on how we could optimize the quality of Indonesian coffee. So first off, I would like to share the story on how we used metabolomics in 2013 to authenticate kopi luwak. And this was very well received by the mass media. In fact, CNN and BBC UK did an interview about this research. And then we showed that metabolomics were very important and highly correlated with coffee quality. We found a particular marker that is related to coffee quality, which is galactinol. And then we also compare many different kinds of coffee coming from different regions. And then we found the unique characteristic of coffee related to its metabolite marker that is unique for coffee in Java, coffee in Flores, Sumatra, and etc. And each country has unique characteristic of metabolites, and we could use that to discriminate and describe our coffee in a more quantitative manner. So here, this is an example on how we could profile coffee from all over Indonesia. And there's like 3 distinct groups that divide our coffee based on the geographical origin very clearly. And we think that because the production place gives terroir effect, this gives result to different coffee taste and chemical characteristic. And we found that the western part of Indonesia has high galactinol, and galactinol was found to be associated with coffee quality. And in our measurement also, the coffee from Sumatra has highest cupping score. And we also investigate the effect of altitude, the effect of post-harvest processes, and we found that post-harvest process was very, very important, the most important variable to change the coffee quality. Then we would like to take all of this information for us to improve the quality of Arabica coffee beans. So fermentation is a very useful method because we could do it very easily and we could optimize it in the laboratory. And we use the bacteria that we isolated from luwak gut, and we optimize the enzymatic activity of these bacteria. We screen them, and then we optimize the inoculum for second fermentation. We apply our different inoculum for 3 different coffee from different origins, and we found that the fermentation did not significantly alter the coffee characteristic, that it loses the origin characteristic, but it still retains the original coffee characteristic. But once we compare independently, in fact, the fermentation significantly changed the coffee quality. And we found also in our research that our fermentation was useful to improve galactinol concentration, which is a quality marker that we found in other study. We also found that this fermentation process can increase GABA, which is a very good compound to promote reducing stress and to make us relax. And I think if we have naturally high GABA coffee, this is also quite good for advertisement. And most importantly, we demonstrate that this is very useful technology to improve the quality through fermentation. And we could increase the cupping score of coffee from 81 to the highest, more than 85 cupping score. And this inoculum and the process have been registered as a patent in 2019. So next, we would like to also describe about the differences in Indonesian chocolate. So we know that cacao beans are very, very diverse, but no reports at all. Compared to coffee, very, very few researchers dealt with the differences and the diversity of our Indonesian chocolate. So we made a first observation that Indonesian cacao beans are in fact similar like coffee, are very unique and different depending on the origin. And therefore, we would like to understand the information from other origins to make a comprehensive profiling of Indonesian cacao beans. And then we analyze in collaboration with Pipiltin Cocoa, which is an industry to produce high quality chocolate. with ICCRI in Jember. And we found very interesting observation that Indonesian cacao beans, in fact, from Aceh and East Java, have higher intensity of GABA compared to the Sulawesi. And Sulawesi cacao beans have already been reported in 2014 to be very high in GABA in other reports. And then we found other sources that have higher concentration of GABA. And this is now under development to produce naturally GABA-rich chocolate, which is food for health. And if you would like to know, in Japan, this kind of trend to produce chocolate with high health benefits with added value is very, very popular. In fact, we have a lot of chocolates that is GABA chocolate, but the GABA is not naturally existing, but it was added to the chocolate. But this is a very good potential once we know the sources of Indonesian cacao beans that have naturally high GABA. We can then produce GABA-rich chocolate naturally. So finally, similarly, in collaboration with BRIN, we also would like to characterize what is the important good point of Indonesian mango. It turns out Indonesian mango, especially aromanis and gedong ginchu, have very similar taste quality with Miyazaki mango from Japan, which is the world's most expensive mango. One mango can be priced for about $50, one mango. And it turns out several of our cultivars have very similar metabolite profile and taste quality with Miyazaki mango. In addition, we also found that aromanis and gedong ginchu has very high vitamin B3, and this kind of information will make our mango have higher quality and added value, and we could then promote this better in the international market. Hopefully, by this kind of information, we could better advertise our cultivar, and this can improve the price of mango in the market and improve the livelihood of our farmers. And I say about the improving the livelihood of farmers, I think that the research that have been described here have been very useful to generate publications, to help students coming from Indonesia graduate and get their PhD, and also to get a patent. But of course, we would like to have our research to be very, very impactful to this to the humankind and to the society. And therefore, I also established a family company for us to interpret the results that were generated from this study and translate it to a way that we could train farmers, we could train small-scale industry to improve their quality, and then it could be exported later on to Japan and penetrate the world's most largest, most difficult market to target, which is Japan. And if we could already be able to penetrate Japanese market, I think it would be easy to penetrate the market. So this is a result of a triple helix collaboration export of Indonesian coffee, cacao, tempeh, and various fruits. We collaborate with industry. We were supported by the government as well. And then through the research of academic, this was finally realized to supported by the Ministry of Trade. Using the information that I have already obtained of the diversity of Indonesian coffee and cacao, this facilitated the export of new origins, new single origins coffee to Japan. And it was realized we take the coffee directly from farmers and buy them directly with higher price. And later on, I hope that this kind of result will really, really translate to improving the standards of our coffee quality. But not only that, we could then realize many different kinds of good commodities for export and penetrate it worldwide. And with that, I would like to close my talk by thanking all of my students, my research team, and all my research collaborators from ITB, from IPB, from BRIN, Pipiltin Cocoa, and ICCRI for their very kind collaboration in order for me to successfully conduct the studies that were described here today. Okay, wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Ibu Sastia Putri, for your informative speech. And Excellencies, ladies and gentlemen, to honor our special keynote speaker, we will have certificate of appreciation that will be given virtually by the chairman of the Research Organization for Agriculture and Food. So please welcome Ibu Puji Lestari, PhD, Ibu Sastia Prama Putri, PhD, and the host to share the certificate. We would like to express our deepest gratitude for your coming at this conference And thank you, Ibu Sastia Pramaputri.
Thank you very much. Terima kasih banyak Ibu Puji. Terima kasih banyak Bu
Terima
Ana.
kasih. Your presence here is very important. So thankful for your availability to share this knowledge to us. Ya.
Thank you very much.
Terima kasih ya Mbak. Terima kasih.
Ya, sama-sama Ibu. Terima kasih banyak Bu.
Ya, sukses ya.
Okay, thank you, Ibu Sastia Pramaputri. Excellencies, ladies and gentlemen, before we continue our agenda, we will have the documentation session. And please kindly all of the participants to open the video and the host to take the picture. Okay, please the host to take the picture. On the count of 3, please. Please all of the participants open the video. Okay, we still wait.
Okay, 3, 2, 1.
Nice.
Thank you. Excellencies, ladies and gentlemen, let's move to our main agenda. Let me inform you that today's conference, we will have 2 plenary sessions. And therefore, I kindly invite Bapak Khairuddin, PhD, to facilitate our 3rd plenary session. Before Bapak Khairuddin start his session, I will introduce about our moderator first. Bapak Khairuddin obtained his bachelor degree at Soil Science, IPB University, and continued his master degree in Food Science and Technology at the University of Queensland, Australia, and doctoral degree at the same university in the field of food science. His research interest is valorization of agro-industrial byproducts, nanotechnology for agriculture and food application. And his current research for 2022 until 2023 is production of biogenic silica nanoparticles from rice husks for biomedical application. And without any further ado, Bapak Khairuddin, PhD, please kindly, time is yours.
Thank you for the nice introduction. Good morning, Honorable Chairman of Agricultural and Food Organization, Head of Research Centers, distinguished speakers, participants, ladies and gentlemen, depending on where you are joining us from. Welcome to the 3rd plenary session at the 1st International Conference on Food Agricultural Sciences 2022. My name is Khairuddin and I will be moderating this 3rd plenary session. In this session, we will be hearing presentation from 3 respected speakers on this very timely subject. But before we start, I would like to make sure the 3 speakers have joined this session, and I would like to take one by one. The first speaker, Ibu Dr. Ratih Pangestuti, are you with us? Ibu Dr. Ratih Pangestuti?
Yes, I'm with you, Pak Khairuddin. Thank you.
And okay, thank you. The second speaker, Ms. Sonita Baba, are you with us? Ms. Sonita Baba?
Maybe we still wait for her. Bapak Khairuddin, you can continue with other speakers.
Okay, so we will be waiting for her attendance. And the third speaker, Bapak Master PhD or his representative? Bapak Master, are you with us? Other representative? Not yet, I think, but yeah, we have to continue this session. Okay, now I would like to share some housekeeping rules to make sure our session today will run smoothly. This 3rd plenary session will be finished at 10:10 Jakarta time, and each speaker will have 20 minutes to deliver the presentation. And I will remind each speaker when the speaker has 3 minutes left to complete the presentation. And to all of us, I think please be active. And should you have any questions for the speaker at any point, please raise your hand or leave your question in the chat box. And please mention to whom the question is directed. And then I will address it during the question and answer session as time permits. Okay, now I'd like to briefly introduce our first speaker, Ibu Dr. Ratih Pangestuti. She is now a senior researcher at Research Center for Food Technology and Processing, National Research and Innovation Agency, BRIN, and she received her PhD from Ukyong National University, South Korea. Her research interests are neuroprotection, bioactivity, marine algae, and nutraceuticals. In this session, she will deliver a presentation entitled Potential Development of Marine Foods in Indonesia. Without further ado, I would like to invite Dr. Ratih Pangestuti to deliver her presentation. Ibu Dr. Ratih Pangestuti, time is yours.
Thank you very much, Pak Khairuddin, for the kind introduction. And I would like to share my screen. Is it clear?
Yes,
okay,
yes.
You can
please
get
continue. Yep.
Yeah, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning, Excellencies, distinguished delegates, speakers, ladies and gentlemen. Welcome to the third plenary session of the ICFAS conference. First of all, I would like to extend gratitude to the chairwoman of Research Organization for Agriculture and Food, Ibu Puji Lestari, PhD, Chairman of ICFAS 2022, Bapak Tri Puji Priyatno, PhD, and also Director of Research Center for Food Processing and Technology, Bapak Satrio Krido Wahono, for inviting me to join at this conference. Well, we have heard the extraordinary presentation from the distinguished speakers, Dr. Sasya Pramaputri, about metabolomic application for food quality and food loss. And today I'm going to be talking to you about potential development of marine foods in Indonesia. I'm Ratih Pangestuti, research scientist from Research Center for Food Processing and Technology, BRIN, Yogyakarta. And let me start first of all by making it clear that food is not an option. Hunger is also not an option for a growing percentage of population. And experts estimate that food production will have to increase by 70% to keep pace with the population growth. And currently, there are almost 60 million more undernourished people than in 2014. And food insecurity has consistently increased since then. And 3 years ago, the number of hungry people grew by 10 millions. And most of this increase has come from Asia, where the majority of undernourished people live. But Africa hungry population is also the fastest growing and currently stands at 250 millions. And there is also the complicating factor of obesity. Just as the world is getting hungrier, every region is also becoming more overweight. The ocean has a great untapped potential to help feed the world, and this resource can be realized with a lower environmental footprint than many other food sources. As the global population and people income rise, the demand for marine food will continue to grow. Fish and plant production from the sea has increased over time, providing an important food source for many across the globe. Marine foods play an important role in global food provision. The number is even higher in some developing regions, including Indonesia and then Sri Lanka, and many small island developing states, which derive 50% or more of their animal protein from aquatic foods. And the ocean can play a unique role in contributing sustainable food security based on 3 concepts, including food production, nutrition, and accessibility. And food from the ocean provide essential fatty acid, vitamin, mineral, and other nutrients that are not found in plant source food and other animal proteins. These nutrients are essential for cognitive development, particularly for children, pregnant, and nursing women. For example, a study in Kenya and Malawi report that dried small fish consumption as a part of daily meal can be a viable strategy for combating nutrient deficiency in the first 1,000 days. And how about marine resource in Indonesia? Well, as we know that Indonesia is the largest archipelagic country in the world with enormous ocean resource. We have more than 17,500 islands, and 3/4 of our territory is covered by the sea. Indonesia is also located at the heart of Coral Triangle, the global center or epicenter of marine biodiversity. We have more than 550 coral species, and with almost 70% of the total coral known species can be found in Indonesia. And our country also have more than 3,000 species of bony fish and more than 850 shark and rays. But it doesn't end to coral reef and fish. We still have many more marine species such as seaweed and also sea cucumber. And in this talk, I will discuss more about seaweed and sea cucumber as a potential representative tropical marine flora and fauna for future food resource. And I will start briefly with seaweed first. Indonesia is rich in seaweed species. The earliest report on seaweed in Indonesia waters probably in 1750 by Rumvius, and in 1860, through the untiring effort of Weber van Boos during the Siboga expedition, the phycology has flourished in Indonesia, where more than 500 species of seaweed has been identified. Unfortunately, however, it is only after 1950 that the economic potential of seaweed has been appreciated, when Zonneveld explored the utilization of tropical seaweed. He stated that marine algae is very important in the future economy of a crowded world. And until now, more than 911 species of seaweed has been recorded in Indonesia, which comprise of 201 green seaweed, 564 red seaweed, and also 146 brown seaweed. Unfortunately, when we compare the seaweed diversity and seaweed usage in Indonesia, it is very different with the East Asian country. The ratio of seaweed consumption in Indonesia is relatively low. Many seaweed species in Indonesia are still categorized as underexploited resource, and only few seaweed species has been used commercially, which is mainly used for phycoploid sectors. And the exploration for food, for example, is limited for few species. These species are used for such as kaulerpa. They are used for salad or ura plato in Jepara, rujak bulung in Bali, karangan in Jogja, lawar seaweed in East Nusa Tenggara, or lawa lawi-lawi in Sulawesi. And currently, many seaweed-based traditional food are vulnerable and need to be conserved. But in contrary, in East Asia, Japan or Korea, for example, seaweed become indispensable part of their cuisine and dietary pattern. And when we went to a restaurant in East Asia, we can simply order sushi or kimbap or seaweed soup or seaweed side dish. But in Indonesia, our country, it is a little bit hard to find seaweed-based cuisine in a restaurant or rumah makan, and therefore seaweed is still remain as a boutique foods in our country. In addition to the conservation of seaweed traditional food, development of healthy and innovative food from tropical seaweed are also important. Seaweed-based product can be rebranded into several products such as synthetic meat, a burger, and other seaweed-based bacon, for example. Well, a meat alternative made from brown seaweed has been developed in the United States, and they have prepared jerk and also burger from the combination of the sugar and the brown seaweed. And this meat alternative can also be developed from seaweed from our country because we have a large diversity of seaweed species. So what is inside seaweed? Why are they so interesting? Well, we all know about the land lettuce, but in this slide, we can see the nutritional value of the land lettuce as compared to the sea lettuce or Ulva lactuca or the green seaweed. When we look at this slide, we can see that the nutrition of the land lettuce compared to the sea lettuce is more complex as compared to the land lettuce. The sea lettuce is comprised of 0% cholesterol, but in the same time, we can find the carbohydrates, protein, minerals such as iron, calcium, magnesium, and vitamin, and many more. And many studies have linked that dietary consumption of sea lettuce and their health benefit effects. Last year, my colleague and I investigated the nutritional value of two edible green seaweed, sea lettuce, Ulva lactuca, and also sea grapes or green caviar, Caulerpa racemosa from Indonesia. And we are using the subcritical water hydrolysis process. And We found that these two tropical green seaweed are rich in protein, sugar, and also phenol, saponin, as well as flavonoid. The highest phenol, saponin, and flavonoid value were obtained from the sea lettuce, hydrolyzed at reaction temperature above 230 degrees Celsius. And we found that the major phenolic acid were gallic acid and phenolic acid. And as we test, we had cytotoxic assay. Both seaweed hydrolyzed did not show any toxic effect in the cell lines, and suggesting that both seaweed were safe and non-toxic to be applied in food and other commercial products. And moving on from the green seaweed to the red seaweed, recently our colleague also just published a paper, and then they investigate different seaweed species from different area in Indonesia, from Java, Bali, Lombok, Sulawesi, and many more. And they are using ultrasound assisted extraction to extract the phenolic compound from the red algae. And surprisingly, they found that the highest level of total phenolic compound was found in Capapicus striatum, followed by Euchema denticulatum, obtained by aquaculture as compared to the wild capture species, suggesting that we can modify, we can optimize the level of phenolic acid in the seaweed by using the aquaculture technique. In addition, seaweed is also rich in functional material, including the polysaccharides, also the pigments, peptide, fatty acid, and other secondary metabolites such as chlorothannin, mycosporin-like amino acid. Well, I give you an example of the fucoxanthin. For example, the brown photosynthetic pigment can be found in the seaweed and also diatoms. It has been widely used in food, medicine, as well as the cosmetic. And in our study in 2013, we found that fucoxanthin were able to inhibit inflammation in microglia, a macrophage which can be found in our central nervous system. Fucoxanthin treatment were able to inhibit the release of pro-inflammatory mediators such as necrosis factor alpha, inos, also prostaglandin E2, interleukin-1 beta, and also interleukin-6. And this is the hallmark of the Alzheimer's disease. Not only fucoxanthin, other bioactive compounds from seaweed also have been recognized for their biological activities, such as sodium oligomanate or GV971. Sodium oligomanate is a mixture of oligosaccharide isolated from the brown seaweed and has been used in China for the treatment of Alzheimer's. And sodium oligomanate received its first approval in November 2019 in China for the treatment of Alzheimer's disease to improve cognitive function. It is proposed that it can reconstitute the gut microbiota and inhibit neuroinflammation in the brain as observed in animal models. And not only for Alzheimer's disease, when we talk about the pandemic COVID-19, seaweed also showed potential to provide nutrition to fight against COVID-19. Well, as we know that Japan is one of the first countries affected by the COVID-19, yet the number of deaths per 100,000 people is like much lower than in other countries. Although they are multifactorial, but several studies suggest that the contributing factor may be diet rich in seaweed. And not only due to the nutrition, sulfated polysaccharides such as iodocarrageenan from the seaweed also has been reported to inhibit virus replication. And in 2021, it was reported that nasal spray with iodocarrageenan showed significant efficacy in inhibiting the replication of the COVID-19 virus. In addition, seaweed also can be developed as a safe and environmentally friendly packaging material in the future. And currently, our research group are also developing seaweed-based bioplastic products by using gracilaria due to their polysaccharide content. And we use the extrusion process to synthesize seaweed bioplastic resin and also bioplastic film. Not only restricted to the marine flora seaweed, also I will discuss about the sea cucumber or marine fauna, which are potential to be developed in the food sectors. Among marine organisms, sea cucumber is an interesting natural source of novel functional material with biological activity that could be used in food as well as by medicine industry. And sea cucumber are soft-bodied marine vertebrate from the class of Holothuredia. And sea cucumber have a leathery skin and elongated body containing single branch gonad. This organism consists of more than 1,000 species, and we can find around 350 sea cucumber species from Indonesia. And we must be proud that, based on history, Indonesia is the eldest and major sea cucumber exporter in the world. Sea cucumber also rich in functional materials such as mucopolysaccharides, amino acid, glucosamine, and many more. And when we browse online, the number of scientific papers focusing on the nutritional value as well as health benefit effect of sea cucumber also increase annually. We can find a large number of studies focusing on the health benefit effect of sea cucumber. And in 2018, we published a review article focusing on the medicinal value of the sea cucumber from the Asia. And surprisingly, we found that many literature demonstrate the biological activity of sea cucumber, including antioxidant, anti-inflammatory, anti-cancer, neuroprotective, and many more. And since it harbors many biological activity, the sea cucumber also called as ocean ginseng or Haisen in China. And unfortunately, the price of the sea cucumber is also highly valuable, like having a large variation. The price in the marketplace are very depending on the wide range of factors, including the species, including the size, but a common notion is that bigger sea cucumber are worth more, and this is often shown in the price that trader offers to the fisher. And then for a given species, the most important factor governing price was the body size. The analysis also indicate that 93% chance that physical damage to the product affect the sea cucumber price. Damage to the sea cucumber could arise from the poor post-harvest handling practices, such as leaving the sea cucumber in the sun before cooking them, transporting them in abrasive container, or handling roughly. Using improper implement to steer the sea cucumber or remove them from cooking pot or soaking them for too long, it also can cause damage resulting in a broken outer body wall of the final product. And therefore, post-harvest treatment of sea cucumber are very important to be developed. And this is the example of correct and incorrect post-harvest treatment of several sea cucumber species, including Holuturia fuscopunctata, Holuturia scabra, and Actinopiga mauritiana. And this is the sea cucumber we have developed in BRIN, including the dried sea cucumber and also rehydrated form of the sea cucumber. And finally, me and my colleagues find a way to improve the quality of tropical sea cucumber from Indonesia. There are at least 5 important steps during the sea cucumber post-harvest processing, including cleaning the chalk layer, boiling, viscera cleaning, salting, and drying. And this is my partner who is developing the sea cucumber post-harvest treatment. And this is some example of the sea cucumber commercial product. We can find the sea cucumber in a dry form, rehydrated form, jelly, and also capsule, canned sea cucumber, and also the intestine form of the sea cucumber called as konowanta. And not only develop as a food, but we need also to develop the aquaculture technique for the sea cucumber and also seaweed and also other marine flora and fauna in Indonesia. So like for example, we can develop the sustainable integrated multi-tropic aquaculture, which can be done as one of the suitable method to be developed, because IMTA will give a better productivity, environmental friendly, and sustainable. In addition, the farmer can harvest the fish for the family consumption and also harvest the other commodities such as milk fish and also the seaweed at the same time for the family income. In addition to the IMTA technique, it is also important to develop the culture of underexploited marine resources such as maybe seaweed and also the other sea cucumber species, because if we develop the culture of the underexploited marine resource, and then
Ibu Rati, you have 3 minutes left.
Thank you, Pak. Because the demand for the marine underexploited marine resource in Indonesia are growing, and therefore aquaculture technology need to be developed so the raw material can be available to support food resilience and marine industry sustainably. And finally, towards sustainable marine food exploration, the achievement of sustainable development goal can be achieved with the development of marine foods. For example, SDG 14, SDG 1, SDG 2, SDG 3, because like if we are realizing the potency of marine food, the nutrition and the health benefit effect, and marine organism can give a better nutrition, which contribute in SDG 2 and SDG 3, while the majority of gender who work in the post-harvest treatment and also product development in coastal area is women. So if we develop the marine food in Indonesia, we can contribute in SDG 5 in gender equality. And by developing biodegradable material from marine resource, such as marine food, we can contribute in SDG 4, SDG 12. And by using the marine organism for carbon sequestration, absorb pollutants, and marine organism can provide solution for SDG 14 on life below water. And finally, development of tropical blue food-based innovative and healthy products is also very important to be developed in our country. And sustainable intensification of tropical marine aquaculture, especially from underexploited resources, are very important. And finally, last but not least, we need to increase the marine food awareness and branding in Indonesia. And before I end my presentation, I would like to extend gratitude to my team, my research group, and my family in BRIN, the Potential Marine Food and Processing Research Group. We are a 14 member coming from different affiliation, background, and age. Let me introduce Prof. Joko, Pak Bakti, Bu Hilda, dan Pak Tegar. And also Pak Dedi, Bu Dipa, Bu Putri, Bu Asih, and many more. And we hope that our research group can give a significant contribution in marine foods developing in Indonesia. And we are very welcome for potential collaboration. And well, I'm sure that I can only deliver a little about tropical marine foods, but I do believe that marine foods can be the solution for the future. And marine food in Indonesia are still undervalued up to now, but I also believe that the number of marine-based food and innovation will grow per year. And therefore, I will say that marine resource can be the solution for the future. However, it's still undervalued up to now. And thank you very much for the chance. I hope we can have a collaboration in the future. I give the time to the Pak Khairuddin. Thank you very much.
Thank you very much, Bu Rati, for the very nice and interesting presentation on potential development of marine foods in Indonesia. Very enlightening for us. Distinguished speakers, participants, ladies and gentlemen, we now move to the next presentation. But because the second speaker, who is Ms. Sonita Baba, here not present, we move to the next presenter or speakers, Bapak Master PhD. And I'll briefly introduce his short CV. Bapak Master is now the president of the Executive Board of Indonesian Biotechnology Consortium, or IBC. Bapak Master earned his PhD from Tokyo University of Agriculture, Japan. His major research interests are about bioregulation and aquatic biodiversity. Bapak Master PhD already prepared a pre-recorded video of his presentation entitled Current Status of Agricultural Biotechnology in Indonesia, Research, Regulation, and Commercialization. Without further ado, please play the video.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Thank you, Chairperson, distinguished participants, ladies and gentlemen. On behalf of KPI, I would like to express gratitude for this chance as invited speaker. I would like to present materi current status of agricultural biotechnology in Indonesia, research, regulation, and commercialization. My name is Mansur from Indonesia Biotechnology Consortium. This presentation is also prepared by Dr. Ika Rustika, Dr. Joko Santoso, and Puji Lestari, PhD. My current priority, besides President KPI, I'm also Director of ICFAS. There are two big issues: first, global food security, and second, climate change. There are some important factors should be get attention. Genetic resources are fundament of agriculture. Very important. Traditional genetic resources contribute to food product and agricultural product. But actually, in some developed country, genetic resources contribute significantly to economic benefit from various products of GR, such as personal care, botanical, biotechnology, and pharmaceutical. Conservation of genetic resources took place in situ, such as agricultural forest and traditional farmland, and ex situ method through gene bank, indoors and outdoor gene bank. Gene bank is important method for agricultural conservation. However, we need big effort in other collection, excellent in quantity, quality, and diversity. Based on quality, total of collection around 30,000 accession, far away from enough. CGIR accession more than 700,000, and some big countries in Asia more than 100,000 accession. Therefore, in quantity context, we should raise our collection more than 5 times. Indonesia has Genome Center in Ministry of Agriculture, Indonesian Agriculture Genomic Center, BGPI, which presents some genomic data of animal, perennial, animal, and mangrove. Genomic sequencing and marker-assisted selection. I would like to explain about genome sequencing and marker-assisted briefly as basis for our discussion. NGS platform contribute to genomics progress using several approaches as de novo assembly, sequencing, transcriptomic, epigenomics. Genome sequencing initially generate read genetic information and determine other of basis ETGC with further studies for either functional, structural, and comparative genomic. This genomic is essential to accelerate genetic crop improvement based on analysis such as genomic size, genomic number, gene mapping, gene sequencing, evolution of crop plant, gene cloning, identification of the marker, MES, transgenic breeding, linkage map, QTL mapping that could be beneficial for important goals including food security, environment conservation, human health, and harness natural energy. This is example of promising genomic method for finding candidate genes for economic traits, especially how to identify candidate gene related to QTL, expressing profiling or mapping population in the mRNA level via molecular analysis to identify expression of QTL for specific cDNA and therefore genes. Correspondent within AQTL peaks for specific cDNA and AQTL peak for a trait causally linked to the function of protein encoded by cDNA provide circumstantial evidence supporting the role of cDNA as a candidate gene for this target traits. This genomic selection commonly applied for genetic improvement livestock crops with long breeding cycle. We have to provide training population that characterize their phenotype and genotype in order to create genomic selection model range. Several statistical analysis and software are used in this approach. Genomic selection refer to the selection decision based on genomic breeding value, of which the genotypic information of breeding population is foundation for the application of predictive GEP for selected population. Genomic selection can shorten breeding cycle, increase selection intensity, and increase genetic prediction. I would like to introduce marker-assisted selection briefly, that the genetic marker are linked genetic variant affecting phenotypic change. Benefit of selection using molecular marker, either single marker or genomic-based marker. They can be conducted on seedling, reduce phenotypic selection and breeding cycle. Middle to high throughput genotyping, low to more efficient cost and time. NGS is a rapid development of functional markers that could be helpful for MES to assess breeding. Marker-assisted breakcross selection are provided using high-density molecular detection for QTL. Next, this slide shows several studies of genomic-assisted breeding in Ministry of Agriculture. A rice variety with a high ferrum useful for biofortification was selected using functional molecular marker. Some land tolerant to iron stress were generated using specific gene that can regulate iron absorption and transport in leaf important for photosynthesis. Marker-assisted backcross was able to produce promising rice line resistant to brown plant hopper and release named Bionic. Integration of PUP1 was successful into new rice variety through MABC to increase the tolerance of P deficiency. Other studies where marker resistant selection on child pepper, resistant on attractors, long juvenile soybean, maize tolerant to drought. Next, in vitro technique. Next, in vitro is branch of biotechnology which are essentially and plant physiology principle, especially related to plant growth regulator, nutrition, growth and development of cell, tissues, and organ, and other mechanisms. The most important function of the in vitro technique is production of seed culture through organogenesis or somatic immunogenesis. Seed of banana, potato, sugarcane, and orchid popular produced through tissue culture. In vitro technique is important method for plant conservation, especially for recalcitrant seed or rare seedling on and flowering plant such as cassava, banana, ginger, sugarcane. On farm conservation requires space land and cost cultivation, so that in vitro conservation is recommended as an alternative. In vitro technique of cryopreservation is standard method for long time preservation, low space, efficient, cost, and labor, and also low risk somacron variation. This method uses nitrogen liquid at temperature minus 196 centigrade degree. Important of in vitro function is for breeding. Mutation in vitro technique conducted at Eka Biograd were created improved variety of sorghum bioguma 1, bioguma 2, bioguma 3, and rice biosalin 1 and biosalin 2. As we know, chromosome pair of crops are varies from diploid, triploid, tetraploid, and also and polyploid. One of important chromosome variation is triploid in this type seedless. Flower biology and pollination resulted seedless fruits at endosperm such as citrus. Citrus was created through endosperm culture. Protoplasm function is also type in vitro technique with ability to integrate to get combination of character two different varieties of citrus. Citrus tangerine, Citrus nobilis, has high productivity, and citrus mandarin, Citrus reticulata, has fresh, more preference, but low productivity, jointed both characters to new improved variety. Genetic engineering. Current status of genetic engineering and genome editing research in Biograd. The application of genetic engineering technique create a great opportunity for crops improvement, particularly for biotic and abiotic resistance. In Indonesia, especially in Ika Biograd, research on plant genetic engineering started since 1097. GM rice efficient for nitrogen use, GM potato resistant to late blight, and genetic on the tomato resistant to tomato yellow, leaf curl virus, and cucumber mosaic virus has been successfully developed by Ika Biograd. Those GM crops have been tested for their resistance to greenhouses of biosafety containment, and confined field trial. Potato resistant to leaf blight caused by Phytophthora infestan. Initial success of GM crop that was conducted by sugarcane tolerant to drought was conducted by PTBN 12 and released in 2013. Based on information from head of Sukasari Sugarcane Experimentation, performance of GMP is better than its control, PL variety. Moreover, Ika Biograd has developed GMP potato containing the RB gene derived from wild relative of potato Solanum bulbocastanum for resistance to Phytophthora etc. The GM potato has released by Ministry of Agriculture as Biogranular Agriculture in 2020. Besides domestic, it was released 4 GM variety of maize last year by 2 commercial seed industry in Indonesia. Therefore, at the present, there are 6 GM plant variety. Moreover, GM variety will rise in number due to hearing conducted in 7 and 8 November 2022. There were 5 GM varieties proposed to release by 2 seeds commercial industry. Minister of Agriculture attended at the hearing to give support for growing commercialization to strengthen food security and agricultural development in Indonesia. Genome editing research. Recently, the emergence of genome editing technology has revolutionized field agriculture. CRISPR-Cas9 is the most advanced and widely accepted because its simplicity, robustness, and high efficiency compared to other techniques such as GFN talent. CRISPR-Cas9 system offer new opportunity to develop improved crop variety with clear-cut addition valuable traits or removal of undesirable characteristics. ICFAS have studied targeted mutation of OSGA to OX2 gene in rice using CRISPR-Cas9 with expression of 2 gRNAs. Gemini virus are among the most devastating pathogens worldwide, causing heavy losses on some crops, including pepper. Knock out the genes related to host pathogen interaction by using CRISPR-Cas9 technology are expected to generate pepper resistant to Gemini virus. Status of research are obtaining the CRISPR-Cas9 construct vector for gene editing and establishing a regeneration protocol. HLB considered is the most destructive citrus disease worldwide and caused by Candidatus liberacter asiaticus. Silencing of gene related to the callose deposit will be a promising strategy to reduce severity of HLB symptoms. Status of research are obtaining CRISPR-Cas9 construct vector for gene editing and establishing regeneration protocol. This slide shows regulation on genetic product GMP list. As we know, the basic convention related to GMP is UN CBD and Cartagena Protocol and ratification law. The Cartagena Protocol is specific for GMP, which ratified under the law 21 in year 2004. Based on this law, released government regulation number 21 years 2000. Regulated comprehensively on biological safety on GMP. DGA regulated the Institution Authority for Assessment and give recommendation for biosafety, Biological Safety Commission, BSC. The chairman position under and responsible to President. The chairman of BSC is assisted by 3 vice chairman, supported by technical team of biosafety assessment, GMP for food, National Drug Agency, for feed, Ministry of Agriculture, and for environmental, Ministry of Environment and Forestry. TTBS leads by coordinator whose member of experts at several ministry, institutions, and universities. Firstly, I would like to mention how procedure to get biological safety certificate. Initial procedure, proponent should propose to Food and Drug Agency for Food Biosafety Certificate, to Ministry of Agriculture for Feed Biosafety, and to Ministry of Environment and Forestry to Environmental Biosafety. BSC received proposal will be asked for TTBS relevant to conducted assessment according to type of biosafety. Result of assessment will be submitted to BSC to be published by Biosafety Clearinghouse for 2 months, and then carried out a completed public hearing. Passed through GMP public hearing, it will be continued by BSC recommendation to give biosafety certificate relevant. GMP biosafety certificate is required to distribution and commercialization of food agricultural products such as GMP food crops and edible plant horticulture as estate crop and feed to distribute and commercialization. Also, for GMP feed biosafety certificate is compulsory for agricultural product for feeds. Some GMP products such as soybean and canola imported to produce feed and some food products, but they are not planted in Indonesia as limitation of climate and other growth factors. However, some of GMP maize event are imported and planned to plant it. Regulation for planting GMP plant are variety released after holding GMP environmental certificate. MOE regulation number 38 2019 provide procedure for releasing GMP and non-GMP plant. Moreover, monitoring and control of GMP plant variety was determined by MOE regulation number 50 2020. The environment biosafety certificate is also standard requirement for decision and commercialization vaccine and drug. So, comprehensive scheme of agricultural GMP types and arrangement under MOE regulation number 38 2019, their last year on 1 September 2020 was hearing and passed to recommended releasing for GMP variety from 2 proponents. Then last 7 and 8 November 2022, it was hearing 5 event GMP maize, which has main character is herbicide tolerant and Bacillus thuringiensis tolerant. Both as institution were backed up by a team of GMP plant valuation and the team for inspection GMP variety. Nah, commercialization. We enter commercialization GMP. If in regulation, we discuss only for GMP as non-GMP such as in vitro technique and marker selection by technology are no regulation. In this part, firstly, I would like to show result of non-GM biotechnology research, especially in IKB grad MOA. Generally speaking, majority of biotechnology improved variety were provided by IKB. How biotechnology technique applied IKB can be shown at the figure. In vitro technique radiation mutation are most popular and dominant technique in vitros. Specific techniques such as hybrid somatic and independent culture were applied for citrus and other culture for improved rice variety. GM molecular technique dominant is marker-assisted backcross. The most adopted in vitro technique are in vitro radiation technique for sorghum and grass. For improved variety are spread to several provinces and growing. The sorghum, Bioguma 1, 2, 3 are very popular and potential for provide food, feed, and fuel. GM plant of maize and soybean are dominant in Indonesia. Both of commodity are foodstuff and feedstuff, especially for poultry industry. However, as different growth ecosystem, which is maize in tropic and soybean in temperate area. Therefore, maize adaptable well in Indonesia. However, soybean is sensitive to photoperiodicity. Therefore, maize is also planted in Indonesia, but soybean is not planted. Last year, GMP maize plant was released for growing and seed produce, distributed and commercialized. Second session GMP maize hearing for release variety was conducted 7 and 8 November 2022. Domestic GMP plant of sugarcane drought tolerance and potato light blight tolerance under process for seed propagation. Commercialization of GMP products and of maize and soybean are extensive sebenarnya through various product of soybean such as tahu, tempe, kecap, soy milk, snack, and other, and feed, and also various product of maize such as snack, flour, and feed. Ministry of Agriculture has support to development of GMP in Indonesia. The minister visited hearing session GMP variety releases, guest visits to support it. We also waiting for sugarcane tolerant to drought and potato for commercialization. We expected adoption of biotechnology GMP and non-GMP will promote food security and agricultural competitiveness. Finally, vision for future agriculture, fund resiliency can be realized. Conclusion: biotechnology need to be strengthened by high education and qualification research, facility, budget, and proper research policy ecosystem. Adoption of biotechnology in food and agriculture research policy will be able to cope with climate change and global food security challenges. Agricultural biotechnology non-GM, such as in vitro technique and marker-assisted selection, should be promoted more to accelerate and improve quality the agricultural invention and innovation, especially through new improved variety. At the present, there are 6 GM plant variety release. This achievement should be increased and improved through acceleration and improvement of effectivity of genetic engineering research. Moreover, we have to take opportunity in raising the number of GM new improved variety through international and seed industry collaboration. Research in genetic engineering tend to move from transgenic to be genome editing. Genome editing research should be given more priority in food and agriculture research program due to several benefits and ability to solve problems, especially related to biosafety risk and regulation mechanism simplicity. Thank you very much for your attention. Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Bapak Master PhD. for a very interesting insight on current status of agricultural biotechnology in Indonesia. Ladies and gentlemen, we are very happy because our third speaker, Ms. Sonita Baba, is already with us. Ms. Soni, are you there?
Yes, thank you so much.
Okay, thank you. Before we continue with her presentation, I would like to introduce her briefly. Ms. Sonita Baba is currently the Biotechnology Affairs Director, CropLife Asia. She holds a master's degree in agriculture from the University of the Philippines in Los Baños. Her current works are related to policy advocacy, knowledge sharing and management, and technology transfer. Without further ado, please welcome Ms. Sonita Baba, who will be speaking to us on plant biotech crops for climate-smart agriculture, benefits, opportunities, and challenges. Ms. Sonita Baba, time is yours.
Okay, thank you very much for the kind introduction. May I share my slide?
Yes, please.
Okay, where is it? Okay, so let me just, can you confirm that you can hear me and you can see the slides now?
Yes. Yeah.
Okay.
It's clear.
Thank you very much. And I would just like first to congratulate BRIN for this first international conference on food and agricultural sciences, and also for inviting CropLife, the research organization for agriculture and food, National Research and Innovation Agency, for inviting CropLife to be part of this very important conference. And I will talk on plant biotech crops, the benefits, opportunities, and challenges as part of their contribution to climate-smart agriculture. And I just want to start a little bit by introducing CropLife. It is a regional plant science industry association based in Singapore. And we have national associations in 15 countries across Asia Pacific. We are also part of a global federation called CropLife International, which is based in Brussels. As an industry association, our advocacy is around enhancing sustainable agriculture, using agricultural innovations that incorporate nature and technology, particularly around plant technology innovations. In CropLife, we have two major programs, the crop protection side and the plant biotechnology and seeds program. So as you know, crop protection products and plant biotechnology and seeds are under different regulations as per mandated by the respective laws. And therefore, part of a bigger work we have is around regulatory affairs. And once this is used, we also have to think about not just on, introducing it, but how to responsibly use products of crop protection as well as plant biotechnology and seeds. We also have work around the issue on anti-counterfeiting as well as protecting intellectual property rights. And all this advocacy work cannot happen just by ourselves. So we do a lot of partnership across different stakeholders. And we are very thankful to the partners that we have engaged over the years. So, my talk today is much easier because Dr. Master has already talked about the regulation, the plant breeding, what are the initiatives. So this will just be a little bit trying to say that, plant breeders, they have a role. Part of their role is to respond to global challenges, whether it be in climate change or either on the climate change brings about pests and diseases as well. And even for developing plant varieties that can maximize the use of resources like your fertilizer or your nutrient, and water as well. We heard about water use efficient plant varieties, and as well as growing more on less, because it is not sustainable to increase all the time area of production. The challenge is how to grow more on less land. And so again, Dr. Master walk us through about the different plant breeding techniques that have been derived over the years. And my talk today will simply focus on DNA recombinant technology, or transgenics, or what they call GMOs. So in 2020, the ISA published a report that the GM crops adoption in 2019, they published 2020, the adoption in 2019 was 190.1 million hectares. Another data source, this GM Ag Bioinvestor.com, the data for GM crop adoption is 195.5 million hectares, which is a 3.7% increase. Much of that, almost 50%, is planted globally via GM soybeans, followed by GM maize, then cotton, and then canola. But we should also recognize that there are other GM crops cultivated across different countries, like the BT eggplant in Bangladesh, the sugar beet, the sugar cane. We've heard that it is also adopted and commercialized in Indonesia, but as well as in Brazil, and then alfalfa. And in the other side, in your right side, you would see the different countries adopting or growing GM crops. And I would just like to note that in the top 10 countries, for example, 2 out of 10 are developed countries, USA and Canada, while the rest 8 are all developing countries, which show the uptake of this GM technology. And it is good to note that 3 of those countries are from our region: India, China, and Pakistan. All of these are on GM cotton. And globally, the GM maize or GM corn got the most number of cultivation approvals. Now, here is a slide that just kind of summarized the findings of the 3 articles published in a peer-reviewed journal, GM Crops and Foods. These articles were around impact assessment of GM crops in terms of pesticide use, in terms of income, and in terms of carbon emission. The focus of the study was on GM corn, cotton, canola, and soybeans. And as far as the traits, it's on insect resistance and herbicide tolerant traits. It is worthwhile to note that the PG Economic Groups, this is the 15th year of its publication. What makes it different from the others is that it provided information on the accumulated benefits out of the adoption of socioeconomic impact benefits out of the adoption of GM crops. So we see that from 1996 to 2020, pesticide use have been reduced, and it increased global farm income. It provided absolute production, increased production for these crops for food, feed, and fiber, and it also reduced carbon emissions. The details of this, I have put the articles, and we can also provide, this can also be, these are all downloadable, free access. I just wanted to share this slide about yield grains, still on the PG Economics report, because you would see here the farm income gain of the cultivation adopting countries. And so you would see that the gain is much higher in developing countries. And this is about 52% share in terms of the income gain as compared to 48% for developed countries like US and Canada. And there are some encouraging news developments, or you could call it opportunities. So as mentioned, aside from GM sugarcane and potato that were approved earlier, this year, their approvals for varieties of GM corn were granted. And so the next part would be then the commercialization of that, making the technology, the seeds of this GM corn available to farmers. And since it's already December, I am really looking forward to next year. And for India, in October, just this year, a biosafety certificate for environment release was granted by the Genetic Engineering Appraisal Committee of India for GM mustard. This is a publicly developed technology. In the Philippines, we have cultivation approvals also of this year for golden rice and BT eggplant. So yesterday, we attended the launching of the golden rice. In terms of, well, I would just say that instead of golden rice, just to really promote it, it is called malusog rice. Malusog in Filipino is called healthy rice. So that's how it is going to be marketed by a public sector Philippine Rice Research Institute. The other encouraging development in the Philippines this year, it took into effect the revised biosafety regulations, whereby the biosafety permit will remain valid unless revoked. And I think this is similar to Indonesia. And then also for stocks events, the Philippine government regulatory agency has its scientific decision not to have a separate assessment for stocks anymore, especially if it is conventionally bred stocks, and the component single events have obtained previous biosafety approvals. However, there is also, they put in place a voluntary registration system for stocks in case the technology developers, whether it is public or private, would want to register their stocks events. And this one on China, I mean, for me, this is like a big thing because they have finally issued in January this year a lot of measures, administrative measures, how they go into variety registration for GM crops, how they go into crop seed production. These provide guidance and pathways for commercialization for local Chinese technology developers who were granted biosafety certificates for environment release for GM corn and GM soybean last year. And generally, there is continued authorization for GMOs, whether it's used for food, feed, or processing. And this is very important because, as mentioned earlier, when you enter a country, before you commercialize, go into market, these have to have regulatory approvals. And when you import, that means this is an additional supply to meet the food security needs of one country. And that is also a very important aspect of something to really remember. The third and final part of this talk is around regulatory harmonization. At the outset, if we do regulatory harmonization, and this is a positive approach, it could reduce cost and facilitate trade. So just looking at the experience we have on the regulatory authorities in terms of safety assessment for food of the different crops, you would see here that globally corn has been assessed more than 9 times. And you would see that all the other GM crops globally available and that are moved across different countries via imports, they have gotten approval by multiple regulatory agencies. And not only that, when you do regulatory assessment, technology developers submit the same data package. And every time the decision outcome of that safety assessment for food, it's the same. It is assessed as safe. So you would see here, for example, the corn event TC1507 has been evaluated by 24 countries and soybean as 22. And not only that, if you consider the inserted genes as well as the proteins that they express, you would see that these have been assessed and reviewed more than 100 times. And now I thought, what is regulatory harmonization? There's a lot of ways by which it can be done. So let me just go through very quickly. For example, you heard about the Codex Alimentarius. So with using that, usually this is the go-to government, this is the go-to safety assessment guideline that is referenced by the government, and through that interpretation, it would be good to have consistent criteria from the different regulatory governments. Data transportability, some of the countries share the same agroecological environment. So the data obtained either in the confined or contained field trials, data transportability means it can also be useful to in the field trials or in the contained trials or confined field trials in the country where it is being done. Regulatory cooperation to trade through trade agreements. For example, COMESA is a trade agreement in Latin America. So if Argentina, for example, approve the same product for cultivation, and then it will also, the same product is assessed for cultivation in Brazil, which is part of the COMESA, then it will be a streamlined process by which they go through risk assessment for environment release. Safety assessment sharing is ongoing between Health Canada and Food Safety Authority of Australia and New Zealand, Food Standards Australia and New Zealand. So what is being done here is that the technology developers submit a dossier to both authorities, and Health Canada, for example, will examine the molecular characterization or the toxicology part, and then FISANS will study the allergenicity part, and then they will share the outcome of their assessment. So either regulatory agency need not repeat the same assessment that Canada does, for example. In terms of streamlined approval process based on approvals granted by trusted countries, I would cite the example of Vietnam. So Vietnam, if you apply for food approvals, if the application or the event is granted approvals by 5 OECD countries, then you get a streamlined approval process, a shorter timeline for approvals. Mutual recognition, for example, the European Food Safety Authority, once it issued a scientific opinion of this application of the event and deemed as safe, then the 27 member states follow suit. They don't need to undergo a separate safety assessment. Now I talk about the CLI harmonization project. Our wish, our goal is that there will be an alignment in terms of data requirements that industry must submit to respective government regulatory authorities. So in 2021, that's just last year, there were the industry experts. You see the member companies, they have a lot of solid industry experts in the regulatory science. And so they have published and these are available in the Journal of Regulatory Science, different aspects on their recommendations to enunciate further how, for example, recommendations for food and feed safety assessment can be streamlined or how it can be done. And another article was published in the earlier year, 2020, how we can assess for human dietary exposure to newly expressed proteins. So these are some of the recommendations coming out from the industry experts, and based on, of course, the long history of familiarity in assessing GM crops. So this is, I think, my last slide. I've shared the contribution and benefits of GM crops, and also how harmonization can benefit in terms of timely access to innovations, because approvals can happen in a timely manner. And that will also improve in terms of mobilization of resources, in terms, say, for example, from a regulatory agency, how much resources is being deployed just to go through the whole regulatory safety assessment review. And as I promised, that would be the last, and I would be happy to provide more information. That's my contact number. Thank you very much.
Thank you very much, Ms. Sonita Baba, for your presentation. It's really addressing our topic today. Ladies and gentlemen, now we come to the question and answer session. We still have about I think around 30 minutes here for discussion. I think there are no one raising hand, but we have a lot of questions in the text box. Okay, we'll try to address the question, but please, I think the first one, the first questions directed to Ibu Rati. Yeah, there are some questions directed to Bu Rati in the text box. Bu Rati already tried to answer the question, but I think it's better if Bu Rati answer directly through this Zoom meeting. I think Bu Rati, please welcome to answer some question directed to you. Bu Rati?
Yes, Pak, I'm sorry, I cannot hear the question. Can you repeat, please?
Oh, okay. First, the question from there is question from Bu Rasiska. Yeah, Indonesia is a maritime country. It's about so. Can you give please give details about the territory where Sika cucumber are being developed in Indonesia, and is there any like commercial company develop this kind of sea products? Ibu Rati? Ibu Rati still with us?
Sorry, Mr. Moderator. Bu Rati said the connection is a bit lost there.
Oh, okay.
Because
okay. Okay. Now we go to next question directed to Ms. Sonita Baba. There are some questions here. The first one, wait a minute, from Ibu Risa Antari. Actually, okay, I try to generalize the questions. Probably, what are the challenges and key strategies to successfully develop GM crops globally? I think we know that generally global communities are still worried about the safety of GM crops, especially their long-term negative impacts. Could you please, address this question, Ms. Tonita Baba?
Thank you very much for the question. So as I have kind of presented, the GM crops are in the market, commercialized since 1996, and until today, they have been assessed as safe as like conventional food, and these are done through regulatory agencies looking at all the aspects on food safety. And there is no incident so far, and I believe there will be none. I am confident about our regulatory authorities' assessment. And also, you don't want to bring products in the market that had food safety concern. The starting point of food safety is always from the developer of the product, and you don't want to go through making that product available if it is not safe. So I believe the opportunity for having more GM food crops or GM crops in the market, just looking at the presentation earlier, the many GM crops that are still in the R&D in Indonesia. So it would help very much. For example, you have regulatory assessment, regulatory harmonization, just how you could streamline the data requirements, noting that those events maybe, I'm not familiar with a specific event, may have been gotten approval by other regulatory agencies elsewhere. And I think we all this familiarity in terms of safety assessment, our regulators have become very expert on this. So I think that is a major hurdle from a regulatory perspective, because for industry, there is a study that says it takes about 13 years to bring the product to market, and more than 60% of that is to hurdle the regulatory science part. So including, of course, getting regulatory approvals. The R&D is there, but there are so many data requirements needed for that safety assessment. And so it would help very much from a public sector perspective, because we have very limited resources. It's sometimes year to year. I work with government before, and so I know that challenge. Funding is always a challenge. Thank you.
Okay, thank you very much for your answer. I think there is a continuation of the questions. Is there any successfully cultivated GM crops in the Philippines?
Yes. GM corn, for example, we have started the cultivation in 2003. So in 2002, all the regulations, as of December 2002, all the regulations are in place. So by 2003, our farmers were already able to access seeds of GM corn, and that have continued. So in the beginning, it was a single trait, insect resistance, but in 2006, the technology developers were able to introduce herbicide-tolerant trait. And I think 98% of the adoption in the Philippines is stacked GM corn. And as I mentioned, this year it is golden rice and beauty eggplant. So yesterday we learned that the golden rice, malusog, the seed deployment is already done. The first harvest of the seeds of golden rice, so that it can be distributed to the farmers, it is already done. And so what we can expect is that beginning, maybe this year, it's a planting season, beginning 2023, you will have golden rice planted in farmers' field in priority provinces. Why do we say priority provinces? So field rice strategy is to deploy malusog, golden rice, to provinces in the Philippines which have the highest incidence in vitamin A deficiency. So that is, and as far as PT eggplant, we understand that seed production is ongoing. Both products are commercially are developed by public sector. Golden Rice is a collaboration between IRI and the Philippine Rice Research Institute, while the PT eggplant is developed by the Institute of Plant Breeding of the University of the Philippines, Las Bañas.
Okay, thank you very much, Ms. Honey. There are some other questions directed to you, but okay, later on, I'll read it. Now we move to the question directed to Pak Mastur. There is question for Pak Mastur. The first one about new variety or what's it, genetically modified new variety of rice which can reduce nitrogen fertilizer. The question is the new variety already released for commercial use? Because if it is already commercially released, it will be very useful. Pak Trijoko, can you address this question?
Thank you, Pak Dede. On behalf of Pak Master, I try to respond to the question from Budiastir ini. Yeah, she asked about the new variety already released or not. Just for information, nitrogen efficient rice, or we call it the NUE rice, was developed by using a genetic engineering approach. And currently the NUE rice line is still under research and evaluation to obtain or generate the data for biosafety assessment, including the environmental safety and food safety, and also feed safety assessment. So we think the commercialization of NUE rice is still require a long time. There are still several stages that must be done or must be passed, including for getting biosafety certificate, and after that the NUE line can be submitted for release. So I think there is still a long way to go for NUE rice to be commercialized. I think that's all, Pak Dede. Thank you.
Okay, thank you. There is another question for you on behalf of Pak Master. The question from Pak M. Yusron. Could you explain the Indonesian government regulation regarding GMO crops, research and development, and its commercialization? Thank you.
Yeah, again, for a GMO product, hello?
Yes, please continue.
Yeah. For GMO product in Indonesia, we have, we call it a precautionary approach in utilization that product. Also for research and commercial implementing. So in Indonesia, we have government regulation of the Republic of Indonesia number 21 year of 2005, PP nomor 21/2005 on biosafety of GMO. And based on this government regulation, we need to, I said before, we need to assessment for environmental safety, food safety, and also feed safety. Yeah, we have a regulation for that. Of course, also for research and development. I think that's Pak Dede.
Okay, thank you, Pak Trijoko. Ladies and gentlemen, we continue to question directed to Ms. Sondita Baba. There are two other questions. The first one from Pak Agus Haram. Ms. Soni, are you there?
Yes.
Okay, thank you. The question is there any long-term observation and analysis in regards of the effect of GMO crops on consumption on human health? I mean.
Thank you for that question. Personally, I am not aware of any ongoing long-term or any completed long-term study regarding that aspect. And probably there is none because, these have been assessed as safe. So it's just my personal opinion. That's why there's no studies. And if there is a study, the challenge is you published it so that others would be able to look at how the study was conducted so that it can be scientifically peer-reviewed. Thank you.
Okay, thank you very much. Ms. Odi, there is one more question for you from Ibu Indras. Ibu Indrastuti. There is a new regulatory on the SDN1 CE that is not categorized as GM crops with the term and condition. Can you explain about this this issue?
So this is referring to the policy about in the Philippines, right? In the Philippines. Okay, so let me begin by saying that if there is a policy, it begins first with the highest making body that sets the policy. So in 2020, the National Committee on Biosafety of the Philippines issued a policy that products, they use the term plant breeding innovations. So plant breeding innovations, they define the whole techniques that only one of these is gene editing. It is not just gene editing. So they use the term plant breeding innovations. And the policy was, how do you determine if this product from plant breeding innovations is within the existing GMO regulation or not? And so from the policy perspective, they said that if there is no novel combination in the final product, it is not considered as GMO. So that's the overarching policy. And then this year, was it March or May? It also tasked the Department of Agriculture. Okay, DA, you set the technical guidelines, you make the details. And so the DA make those details. Even then, in the National Committee on Biosafety Policy, when they issued the policy, it comes with a decision tree analysis where it says, these are the techniques. Are they GMO? Do they contain novel combination or not? And under that flow chart, SDN1 is listed as non-GMO because it doesn't make any novel combination. And so SDN1 products, but they put in place, the Department of Agriculture put in place that if you believe that you have a product from SDN1 or whichever, SDN2 or 3, and you want the government to determine whether your product is a GMO or not, they put in place a process for technical determination. Then there is a procedure. They all put that together, and that involves a lot of consultation, not just the technology developers, both public and private, but other researchers and other policymakers, even the government policymakers not involved in regulation, because they have to understand. And so SDN1 is not considered under the GMO regulations in the Philippines.
Okay, thank you very much. It's clear. Ladies and gentlemen, for some question directed to Burati, we just received information from Burati. She's having technical problems with the connection. So she will try to answer the question through the chat box. But because we are now running out of time for this session, so any question that cannot be answered in this session, please connect with the speaker one-on-one through a private message.
Yes.
And finally, on behalf of the committee, I would like to thank you very much. I believe we had a great discussion today. Thank you for being very active in this session. Now, I would like to request all of you to give a round of applause to our speakers. Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. And offer the next agenda to the MC.
Thank you. Thank you very much. Thank you.
Okay, thank you, Bapak Hairudin, for the session, and to our wonderful speakers for the informative speech. Excellencies, ladies and gentlemen, to honor our special invited speakers and moderator, we will have certificate of appreciation that will be given by the chairman of the ICFAS, Bapak Tripuji Priyatno, PhD. So please welcome for our first invited speakers, Ibu Dr. Ratih Pangestuti. It is possible to open your video. Bapak Tripuji and the host to share the certificate. So please. Okay, thank you.
Thank you, Dr. Ratih Pangestuti, for your wonderful sharing on marine resources for food. We highly appreciate for your contribution. And thank you to Bapak Mastur PhD for your sharing on current status of agriculture biotechnology in Indonesia, and also for KBI support in this conference.
Okay. Sooner ada ya.
Okay, please the host to take the picture. Okay, we continue with our second invited speaker, Miss Sonita Baba.
Okay, thank you for Miss Sonita Baba for fruitful sharing on plant breeding and biotechnology. And thanks again for CropLife contribution in this conference. Thank you very much.
Thank you so much.
Thank you. And the next speaker, please welcome Bapak Mastur PhD.
Thank you, Bapak Mastur PhD, for your sharing on current status of agriculture biotechnology in Indonesia, and also for KBI support in this conference.
Thank you. And the last to our honorable moderator, please welcome Bapak Hairudin PhD.
Also, I would like to thank you to Bapak Dede Khairuddin for moderating this session. Thank you very much, Pak Khairuddin.
Okay, thank you. Thank you, Bapak Tripuji Priyatno, PhD. Excellencies, ladies and gentlemen, next to our agenda is the fourth plenary session of the conference. And therefore, I will invite Bapak Agung Hendriadi, PhD, the chairman of our last plenary session. But before Pak Agung Hendriadi start his session, I will introduce about our moderator first. Bapak Agung Hendriadi, PhD, is a senior researcher of Research Center for Agro-Industry. He got his doctoral degree from Asian Institute of Technology, Bangkok, in the field of agriculture system engineering. His current publication entitled Household Food Waste Policy, a literature review published in Journal Kesehatan Lingkungan 2022. And without any further ado, please welcome Bapak Agung Hendriadi, PhD. Kindly, time is yours.
Yeah. Thank you, Bu Ana. I think we go earlier. This plenary session. I hope they will finish earlier also, because it is Friday, right? So we will have two invited speaker. In this plenary session. One is Dr. Sunil Arcak. He is from National Bureau of Plant Genetic Resources, and Dr. Bat Parnish from Biodiversity International Laboratorium. Before start the presentation of Dr. Sunil, I will call Dr. Sunil. Are you with us now?
Yes, I'm here.
Okay, thank you, Dr. Sunil. Dr. Sunil is a specialization in plant genetic resources, PGR information, and PGR policy. He has 25 years experience working with the various aspects of plant genetics. He developed many web-based applications, including PGR portal. Dr. Sunil Arcak also represented India FAO ITPGRF FA governing body meeting and Kilaki, Rome, and Delhi. He is also chair the working group of to enhance functional and multilateral system of plant treaty and member of the scientific. He also advisory committee of the Global Information System. He is also representing India in FAO, CGRFA, and CBD meeting. Today, Dr. Sunil will talk about the plant genetic resources policy and governance. Dr. Sunil, time is yours.
Yeah, but please allow me to share my slides. Yeah. Are my slides visible now?
Yeah, yes. Yeah, right.
Okay, excellent. Thank you very much. Thank you, first of all, to the organizers for having me in this particular congregation. And then I have been listening to very interesting discussions before. I am here to talk to you about a little bit different than the actual research. It is about the governance of plant genetic resources. And we have new challenges. Governance has been always a challenge, and we have new challenges. So I'll just talk to you about this particular thing very briefly. I come from National Bureau of Plant Genetic Resources, as has been introduced already, kindly. And we work on all the aspects of managing plant genetic resources throughout the country. The moment we hear the word governance, it appears that it is something like policing, something to stop. But it is not so. Governance is for facilitating. So we have a germplasm, and the governance structure facilitates the access, and the governance structure also facilitates the benefit sharing. So what is the final purpose? Final purpose is conservation and sustainable use of plant genetic resources. So PGRFA governance aims at guaranteeing food security for all the people around the world. But it is not very easy. In principle, it is very straightforward, but in implementing this, it's not very easy. It is a little complicated because there are various issues that are involved in this, such as benefit sharing, how it is directly influencing the agriculture, what is happening to various gene banks, and how do we know what is genetic material, accessibility to food, etc., etc. And we have something new, which is called as DSI. I will talk about that a little later. We have many global instruments that governing the PGRFA, and perhaps you all know some of them already. We have Convention on Biological Diversity with two of its important protocols, Nagoya Protocol and Cartagena Protocol. We also have WTO various agreements. We have International Treaty on Plant Genetic Resources for Food and Agriculture, and a number of countries around the world are signatories for these international instruments. The moment we are signatories for these international instruments, they are legally binding and we are supposed to implement in our countries. So to implement them in our countries, we have our own national institutions. For example, in India, we have National Biodiversity Authority, Plant Variety and Farmers' Right Authority, Patent Office, etc., etc. So the complications start from how do we define plant genetic resources? We can govern plant genetic resource only when we know about the definition of the plant genetic resources and the scope of plant genetic resources. We usually think that plant genetic resources are directly nothing but purpose is the conservation of the material. So the material that we have, and as well as the documentation, the associated information that comes along with the material. However, it is very important to know that for implementing governance structure, we have to define plant genetic resources clearly, as well as we should know what all is involved in the plant genetic resources. That means temporal, from when it has started, the scope of implementation, and the spatial, what is the governance, the jurisprudence, where we can control that one. And after that, we have the other part, that is access and benefit sharing based on both global and national laws. So how do we define plant genetic resources? You and me, all the researchers who are using plant genetic resources, we think that some material that we access, that we use for our research, that we gain from the nature outside, from gene banks, from our colleagues. So all these are plant genetic resources. But it is very important to remember and understand that they come in various forms, and then it need not be always seed. For example, in a single crop like potato, it can be a seed, it can be in different forms. You can see here on the slide. And additionally, in recent times, we also have DNA sequence as plant genetic resource. So this has added a little more complication to the governance of plant genetic resources. We will try to understand. Now, it was not sufficient for just to understand what are plant genetic resources. So people thought that in 1983, in the undertaking globally, they tried to define what are plant genetic resources for the purpose of access and use. So you can see here in the topmost definition, it is reproductive or vegetative propagating material of various categories: cultivated cultivars, obsolete, primitive, wild and weedies, genetic stocks, etc. But that was purely for the purpose of research, access and research use. The moment it started coming to more and more techno-legal purposes, then we had to define the same set of plant genetic resources differently in IT-PGRFA. Look in the last, the box. Any genetic material of plant origin of actual or potential value for food and agriculture. So in the treaty, it is restricted to only food and agriculture, and actual or potential value is added here along with the genetic material. So it is not just the propagating material. The definition now is genetic material, and individual treaties, individual instruments defined for their own purpose. For treaty, International Treaty on Plant Genetic Resources, it says it is only for food and agriculture. However, the same set of genetic resources can be defined for other purposes in Convention on Biological Diversity, including pharmaceuticals, chemical industry, etc. For example, we can have a little bit of changed definition internationally and nationally in each of our countries. For example, Convention on Biological Diversity says biological resources includes genetic resources, organisms are the parts, whereas in our country, in India, biological resources includes their genetic material as well in the definition. So when we want to govern, we want to implement some kinds of rules and regulations and the framework, our definition, Government of India's definition says that it is genetic material as well. Now, how this will affect, I'll tell you a little later. Okay, now all the researchers like you and me around the world work for acquisition of the material. They go and collect, explore around the country, collect very valuable material, they import from other gene banks, and then they conserve it using complementary conservation strategies. And then they carry out lots of experiments so that we know the value of these material. Okay, so once we know this value of the material in terms of their traits, these material are now supposed to be available. They are now available. These valuable germplasm lines are now available. but they may not be accessible. For them to be accessible, there is an in-between, we have governance. So how can these available genetic resources can be made accessible? Look here, for example, we have various forms, various kinds of, let's say, conservations that are going on around the world. We have CG gene banks such as IRRI in Manila. We have CIMIT, we have other gene banks, ICRISAT in India. So number of CG gene banks are there. They do not differentiate between public sector and private sector. They do not differentiate between within their own country or some other country, because these are global organizations. Their purpose is to supply genetic material, the germplasm lines for all the intenters, for all the users, potential users. Whereas national gene banks are governed by their own rules and regulations. They may supply to public sector institutions very easily, freely, but may not be very freely to the private sector organizations of even their own country. And what happens to the material if somebody asks from other countries is much more difficult. And what is happening in community seed gene banks? These are for their own internal maintenance purpose. So materials that are conserved in on-farm, on the fields of the farmers and the communities, and in situ, which is outside in the nature, these may not be directly accessible to all the people. So they have to come through one of these gene banks and then may be accessible to the researchers. So this is how they may be available at various places. Whether they become accessible depends upon what is the governance structure. So how do we access plant genetic resources? For example, the gene banks hold germplasm lines. They also have information in various databases, without which these germplasm lines become useless unless we know the value of these germplasm through their information. And they have various institutions, national institutions, because then those procedures have to be followed. And the rules apply based on whatever national and global rules are there in those countries. This is where some of our governance challenges begin. So gene bank accessions are there in the gene banks. The management support is by the domestic country, the government. Whereas all the indigenous germplasm lines are represented here or not, we are not sure, because the collection and then conservation is not complete any time. Now, assuming that some of these germplasm accessions are there, they have to be now designated for multilateral system so that users can access them. All the crops and accessions are not are not designated usually. Why? Because gene banks are strapped with the funds crunch, and the cost of multiplication and maintenance of medium-term storage is not easy. So everything need not be accessible because logistics and handling charges are prohibitive sometimes. So at the end of the day, the available gene bank accessions may not be accessible to everybody, even within the country. So that's a challenge. Now the challenge increases because the international rules say that for example, in case of treaty, whatever is the benefit, it goes to a common fund. That means the conservation and supply that is being taken care by the national government and the national gene bank, whereas the benefit sharing goes somewhere else. So this decoupling of conservation and benefit sharing creates sometimes problem in implementation. So this is an disincentivization. There are some other complications. For example, look at this picture. All these dots show the sources from where you can access the germplasm of the origin of India. Okay, see, all these gene banks hold material of Indian origin. So that means the material of Indian origin is accessible elsewhere also, other than India's own gene bank. For example, around 63,000 accessions are there in 11 CG gene banks. So it's not that only the country governs within the domestically, they're already available elsewhere. Okay, so the access is one side. Now, what is happening to the benefit sharing? Now, benefit sharing can be even more complicated. Just look at this picture. Okay, this is a couple of years ago. This was published based on a survey. Industrial users, academic users, okay, and then providers. They all have various factors that are influencing the complications of the benefit sharing. Whether there is sufficient predictability, legal certainty is there, transparency is there or not. whether there are sufficient incentives are there for conservation of biodiversity. So all these parts are there. So it becomes little more complicated. Now add to that, what has happened over the years after these international instruments have been signed, such as Convention on Biological Diversity, things have changed drastically in the technological vista. Number of technological developments have taken place in plant biology, in agriculture, because of the advancements in genomics. Now, because of the advancements in genomics, huge amount of DNA sequence data of all these species that are important for agriculture or pharmaceuticals or for life sciences are available in public domain. So what happens to these? People can access, users can access these sequences and can make use of them based on new tools such as gene editing, which you people were discussing in the previous session. Now, what happens to this? Now, I have, let us say, a country is conserving a specific endemic species, which it has huge value for, whereas a sequence is existing in, let us say, NCBI database in USA. Anybody can, from anywhere in the world, can access that one and use that one without any requirement of a permission or a benefit sharing with the original country. So that means we have dematerialized. This is called as a dematerialization of the access and use of the germplasm. So how do we manage this kind of governance structure? So these have serious implications. So what is happening around the world? In treaty, all these, the DSI, this is called as digital sequence information or genetic sequence data variously, because definition has not been finalized, scope has not been finalized. What will happen to this? How do we manage has not been finalized. So what is happening globally? In treaty, in plant treaty, people are working on what is called as an enhancement process of MLS, multilateral system, in which the DSI is also being considered. I am one of chair of this particular committee, and we know that the job in hand is very, very difficult. But we want to make it more and more inclusive. But we can make it more inclusive only when different countries around the world have developed their own position. For example, if Indonesia has developed their position, what Indonesia thinks about DSI, how Indonesia wants to govern DSI in her own country, unless that is not finalized, we cannot have a common view. We cannot have an enhancement process. So it is very important for all of us in different countries to understand this particular thing, to have dialogues, to engage with each other, and then develop our positions properly depending upon our own stakeholders. And this is very important. And in case of Convention on Biological Diversity, people are developing what is called as a multi-criteria matrix. And we have in few more days COP15 to be taking place in Montreal, and where we are going to discuss this. So these are becoming more and more complex. We are adding ourselves complexities to this simple process otherwise. Okay. Are gene banks around the world DSI ready? We know that these things are happening and many more things can happen. Are we ready? If I am a gene bank manager and if somebody else, you are gene bank manager, are we ready? How do we manage? How do we respond to this DSI conundrum? Now, whether we know exactly how our accessions and DSI around the world are linked, do we know about that? No, we don't know. How do we supply germplasm? We supply only germplasm seeds, or we supply along with the information which is DSI? Or what about the feedback by the users? If somebody takes material from me, from our gene bank, and generates sequence, do they have to give me feedback in terms of sequence as well? No, nothing is known. So the governance structure framework for DSI is yet to be developed. It is evolving. So in India, what we have done in our place, we have developed a tool, and this is called as G2G, Gene Bank to Gen Bank, G2G. So what it does is, it tells me all the gene bank accessions that are present around the world in different gene banks by name, by the cultivar name. If there are any DSI, that means DNA sequence, a protein sequence, an RNA sequence, or express sequence tag, EST, what do they call? So if you don't understand all these specificities, don't mind. What is important is the genetic material in terms of sequences, in terms of the things which are not tangible like seeds, are available elsewhere. So we are trying to link both of them. So this tool which we have developed helps us to know that, okay, these are the accessions that are present in my gene bank and their sequences are available. So I have to track these, I have to find out, not just to stop that one, to ensure that facilitation is there and benefit sharing is ensured. Because the material that are present in my gene bank or your gene bank do not belong to these gene banks. They belong to farmers and communities. We are only custodians of this. So it is our duty that the benefit sharing reaches those communities if at all commercial benefits are earned by accessing or using these material. And people are proposing different ways of benefit sharing model for DSI. This is, let's say, one of my colleagues in treaty, Amber Scholz from Germany, and her colleagues have tried to develop what could be a different model, but they're all not easy. They're all very complicated. So to just end my talk, I just want to give this message to all my colleagues listening to me, that we might be working in lab, we might be working in the field, but it is very important for all the researchers using plant genetic resources that you must know rights and responsibilities with respect to their use. We have both. We have rights as well as responsibilities. And there are global instruments. However, they can only set the framework and then how individual countries can work. They can only guide. But the domestic legislation is always overrides. The domestic legislation overrides all these things. So if you have a clear domestic legislation, that is one which overrides the international things. That is the one which has to be implemented. And for all of us, for the benefit of users, for the providers, it is very important that organizations such as gene banks, we create a single window system to facilitate the access of these valuable germplasm lines. Because please remember, despite all these governance structure, national institutions, international instruments, the ultimate aim, ultimate goal is conservation and sustainable use of plant genetic resources for global good. That is the most important point, through research, through education, through awareness. Okay. So I thank you all, and I acknowledge various my colleagues and then funding agencies, and I stop it here. Thank you very much.
Thank you, Dr. Sunil. Very nice presentation. It's very interesting. We talk about the management of plant genetic resources. I believe that there will be a lot of questions. Then I will come to the second speaker, invited speaker, Dr. Bart Panish. Are you there?
Yes, hello. I'm here.
Thank you.
Hi.
Dr. Bart Panish, a senior researcher and of Biodiversity International Living Begin. He is interested in the development of the novel plant cryopreservation protocol. He graduated from PhD from the Leven Belgium, postdoctoral researcher, and later on as the research manager, he coordinated different international projects dealing with the plant biology, biotechnology, and cryopreservation. Bart Panish is currently member of the additional board of the Cryo Letters and associate editor of the PCTOC. He trained more than 100 researchers from all parts of the world on plant cryopreservation and developed protocol for about 50 plant species. He also researched. His research was written down in the more than 400 contributions, resulting in its index of 32. Dr. Panish, Dr. Panish, time is yours.
Okay, I will first share my screen. Okay, can you see it?
Yeah.
Okay. So thanks for the previous presenter to focus a bit on how to share plant genetic resources and the background, the policy. So my name is Bart Panis. I'm linked to the Banana Gene Bank collection in Leuven, the collection which is managed by the Alliance of Biodiversity International and SIAT. And I will focus a bit more on how to conserve the genetic resources. First of all, some facts. So there are many people that are undernourished, as we all know. And also the world population is expected to increase quite considerably during the next decades. You see here the projected evolution of the world population. We see that in Europe and North America, South America, the population remains quite stable. In Asia, the population has increased considerably, but we are expecting a kind of lag phase within the next decades. But you see in Africa, for example, that we still expect a lot of population increase. We are also dealing with climate change. Climate change is causing increased temperatures, difference in precipitation, different carbon fertilization for plants. So this means that in some cases, the climate change will have a positive effect on production. But what you also see from this map is that, in fact, the places where we expect the highest population increase will also have a negative effect on plant production, on food production. So we urgently need some measures there. So how can we deal with this increased world population and the climate change? We have to produce more food. That is quite obvious. How can we do that? We can increase cultivable land, but of course, as you see from this figure, this is also limited. I mean, we have deserts, we have Arctic regions that cannot be cultured, and the land that can be cultured is already in use. So this is limited. So we have to look for alternatives, improve cultural practices, fertilizers, use fertilizers, crop rotation, increase biotic, abiotic stress resistance of our plants, and of course, resulting in an increased yield. But all these measures are depending on the utilization of plant genetic resources, the availability of plant genetic resources for breeding. What is also clear is that in fact we only use a fraction of plant genetic resources to feed the world. There are 250,000 plant species identified. Only 3 of them are providing more than 50% of the world food energy intake. So plant diversity that is used is limited, but also the diversity that is used within a crop species is limited and decreasing. So we have to take care of that. So how is plant germplasm stored? So there are different ways. Of course, we have the in situ conservation, where the material is stored in normal habitat. This can be the natural habitat, for example, in rainforests, but also this can be in farms. And then we have also ex situ conservation, so where you take in fact the material out of the normal habitat and store it somewhere. A very good example, of course, are the seed collections. Seed collections, you take the seed, you dry it down to a moisture content, then it can be conserved at -20 degrees, for example. You see here some examples. In Colombia, you have the BRIN Gene Bank, one of the CG centers. In NPPGR, linked to the previous presenter, you have also big collections of all kinds of food crops. And also a nice example is the global seed vault. It's a tunnel in the permafrost in a small island in the north of Norway, where materials from all over the world is stored for the long term as a kind of backup. You see here where the red dot is, that is where Svalbard is, that seed fault, and the green and the yellow circles represent those institutes that have backupped their materials in Svalbard. So the yellow ones are the CG centers, and the green ones are the national gene banks. But what is the problem is that many of these food crops cannot be conserved by seeds. Why? Because some of the crops, they simply do not produce seeds, like bananas. Edible bananas do not contain seeds. You have crops that not breed true from seeds. Good examples are the fruit trees that are clonally propagated. Apples are grafted, for example. These are clones. If you want to conserve the specific gene combination, you have to store the clone. And finally, you have crops that produce seeds, but from which the seeds cannot be stored. I mean, they are called recalcitrant. So this means that when you dry them out, you will kill them. So for this, we need to look for alternatives. This can be in field gene banks, botanical gardens. Here you see a field gene bank of potato, for example, in IPK in Germany. But then we have also in vitro collections, because field collections, of course, material is still subject to diseases, adverse weather conditions, so you can lose your material. So you can take it also out of the field and put it in vitro. You store the different genetic materials in test tubes. And this you can do under normal growth conditions, where the material grows at a fast rate. But the problem is that you need to subculture. You need to put the material every 2-3 months in a new tube. So this is quite labor intensive, especially if you are having a big collection. Or you can also store that in vitro material at slow growth conditions. This means that it's more slow and that you only need to subculture the material once per year, for example. And then finally, you have cryopreservation. I will tell you more about that because I'm focusing my talk a bit on cryopreservation. And the DNA banks, as already mentioned by the previous speaker, it is interesting, of course, for molecular biologists or for people that want to do gene editing or genetic engineering. But of course, still no plant can be regenerated out of pure DNA. But it has, of course, its advantages. So as I told, the cryopreservation is also an alternative. So what is cryopreservation? Cryopreservation is the storage of biological material at ultra-low temperatures. So most of the time, a temperature of -196 degrees Celsius is taken because this is the boiling point of liquid nitrogen. And why this temperature? First of all, we know that at this low temperature, all chemical biological processes are arrested, so you can store the material for unlimited periods. And secondly, it's also quite practical because you just have to store the material in a kind of big Dewar flasks filled with liquid nitrogen. So you see here 2 examples. One example is the cryobank in NPPGR in Delhi. And here you see some of the cryo tanks we have in Leuven for the banana collection. But of course, in theory, this sounds quite nice. You put material in liquid nitrogen, but you also can understand that not all biological material will survive exposure to such low temperatures. So you have different problems arising there. But the most important and damaging is the injury that is caused by ice crystal formation. I'll explain you why. So we all know what water is. Water are water molecules which are moving chaotically around. And when water freezes, it forms ice crystals. But also you can solidify the water at low temperatures without forming ice crystals. And this is called vitrification. So you see the same chaotical structure as with normal liquid water, but all the molecules stop moving. So this is what is called a vitrified state. And how can you see that? So this is, for example, an Erlenmeyer flask with water. You see it's quite transparent. An Erlenmeyer flask with water that has crystallized, it is white because of the ice crystals, and water that has vitrified. So you see it is as transparent as liquid water, but this is a solid condition. And these crystals have effects on the plant membranes. In liquid, membranes are quite intact. When you have ice crystal formation, you see that the ice crystals formed will penetrate and damage the membranes. So the membranes lose their permeability, semi-permeability, and in a vitrified state, also you see that the membranes are not damaged. In fact, this is what cryopreservation is all about. It is about preventing ice crystals to be formed and killing the cells or the tissues. How can you prevent this? I'm not going too much into detail there, but you have to remember two things in order to prevent ice crystal formation and to initiate vitrification, a vitrified state inside your cell. You need to concentrate your cell solution. This can be done different ways. You can remove water by air drying, freeze dehydration, osmotic dehydration, penetrating cryoplastic substances, adaptive metabolism, so a kind of acclimation. And also you need to apply rapid cooling and thawing rates. In fact, this is to prevent water molecules to organize themselves into a crystal. In fact, you just say to the water molecules, immediately stop moving and you have a vitrified state. So taking this into account, different cryopreservation methodologies were developed. But the ones that are encircled here are the ones which are in practice most used. So what is the state of the art of plant cryopreservation? So how much is it used? So some years ago, we did a feasibility study to ask at different institutes how they use cryopreservation, what is their projection, what are they planning to use it, and these are the institutes to which we send the survey and had the responses from. And the conclusion is that only 17 crops have cryopreserved collections of more than 100 accessions. So you see the biggest collections are potato, apple, garlic, mulberry, banana. But then you have also a few minor, and then you have also some collections where only, let's say, 10 or 20 different accessions are cryopreserved. And what is the material that is preserved? This was also mentioned before. You can cryopreserve dormant buds, but also you can cryopreserve buds or meristems excised from in vitro material, but also pollen, seeds, zygotic embryos, all these kind of tissues can be cryopreserved. And what you cryopreserve depends on the species that you work with. Apple, you can have dormant buds there. You aren't going to cryopreserve dormant buds. But in banana, you use meristems excised from plants. For citrus, for example, we see that a lot of embryos are cryopreserved. So really depending from the species, we cryopreserve different propagules. Here you see an overview of the different institutes that are cryopreserving specific crops, the number of accessions that are cryopreserved or stored for the long term, and also the method that is used. In many cases, droplet vitrification is used, you see, but also dormant bud freezing. Why dormant bud freezing? Because this is the most simple and the least labor-intensive protocol, but you cannot use that dormant bud freezing protocol for all plant species, only for those species that, first of all, produce dormant buds, and secondly, those species that can be grafted. But for the majority of species, droplet vitrification is applied. Now something on bananas. So it's an example on how to preserve the germplasm for a specific species. I talk, of course, about bananas because I'm linked to the banana collection in Leuven in Belgium. So I do not have to explain you the importance of banana and plantains, because I think that Indonesia banana is quite important.
Excuse me, Dr. Barat. Excuse me, you have 3 minutes left.
3 minutes only. Okay, I will go fast. Okay, so it's an important crop. Biggest productions of plantains and cooked bananas and bananas you see here. Indonesia is the 3rd biggest producer of bananas, for example. India is the biggest one. There are many problems in banana cultivation, diseases, stress due to climate change, fruit, and for this we have to make new cultivars. And where do they have to come from? From banana collections. This is an overview of the banana collection in Leuven. We have an active in vitro collection for 1,700 accessions. But we have a backup of this collection in liquid nitrogen, about 1,200 accessions. We have a DNA bank, a lyophilized leaf collection, and the cryopreserved collection is moreover backed in Montpellier. It's about 1,000 accessions which are stored in another place, because suppose something goes wrong with our collection in Leuven, we can always re-establish the collection from materials in France. So we developed a droplet vitrification protocol, which seemed to be quite efficient. It is so efficient that we could apply it to different species in collaboration with many other institutes. We also store wild bananas. So there are different wild bananas which are at the origin of the edible bananas. You see, this is the place of origin. Indonesia is very important for wild bananas. This is how they look like. They are very nice, very colorful sometimes. And what activities are linked to this storage of wild bananas? So we are collecting, we do some collection missions. For example, these are some pictures from a collection mission we did in 2019 in Papua New Guinea. We are looking at the germination of bananas, because banana seeds are difficult to germinate. The germination frequency is very low, but we are optimizing that. We are looking how to store these bananas, different temperatures, different drought conditions. We even try to store bananas in liquid nitrogen. Unfortunately, banana seed is orthodox, so it can be stored. We are looking at population genetics, because we want to know that we really store the populations and not only a fraction of the DNA. We did some species distribution modeling to see where in the world we have to go for collecting specific accessions. And then finally, what we also do is phenotype bananas, seeing wild bananas, seeing, okay, which individuals can provide resistance towards, for example, drought. So the conclusion of my talk, so the conservation and sustainable utilization of plant genetic resources are keys to improving agriculture productivity and sustainability, because climate change, increasing world population. There are different storage methodologies, but what to store depends on the species, the available plant materials and the facilities. For cryopreservation, you need cryopreservation facilities. For long-term conservation, cryopreservation should be considered, but also seeds for vegetative materials, but also seeds can be considered to be cryopreserved. And also it is very important to conserve the crop relatives. Okay, that ends my talk. So thanks for your attention, and I'd be happy to answer any question.
Thank you, Mr. Panish. Very interesting also. We are talking about the strategy plan of genetic conservation, especially we talk of the cryopreservation of banana. Very interesting. Okay. Now we come to the question and answer. Let's see. No raise hand there. Okay, I will start with the chat box. Oh, one raise hand from Mr. Ika Rustika, Miss Ika Rustika. Please, Bu Ika, silakan Bu.
Yes, thank you, moderator. Dr. Bat,
Hello.
nice to see you again. Yeah, thank you for joining the event. It was a complete presentation from you of story technique and also many plans and with image that make easy for us to understand about cryopreservation. I hope there will be a follow-up for us that we can collaborate between BRIN and COOL maybe. And I will ask to you, so many plant species you have received. So in the future, what kind of species will you consider to be cryopreserved? Thank you.
Okay, thank you, Ika. Nice seeing you again. Well, I think so. There are some species where the cryopreservation protocols are quite well developed, and like potato, like banana. I mean, then there's no need for optimization. But there are still some species like cassava, like sweet potato, that need more work to optimize the cryopreservation protocol. But we have to take into account, of course, cryopreservation is a quite labor-intensive task. It requires a lot of investment. Just to give you an indication, for most species, one well-skilled technical staff member can cryopreserve about 50, 60, 70 accessions per year. So this means that it is an investment for the long term to do cryopreservation. So this is something which always has to be taken into account. So you can develop a protocol and you can have technical staff, a student for that. But finally, to apply the protocol, you need quite some staff. And that is something which has to be taken into account. But I think it's a future for the long-term conservation for vegetative material as well as for seeds. I hope that this answers the question.
Okay, Bu Ika.
Yeah, thank you very much.
Okay.
Thank you, Pak.
Now, other question from the. Okay, I will continue with the text chat box. There is two questions to Ms. Dr. Sunil.
Yes.
First is based on the current genetic resources. It's from Madhetasma, Dr. Madhetasma, to Dr. Sunil. Based on the current genetic resources definition globally, are genomic DNA sequence as defined as genetic resources?
Okay.
Yeah.
Yeah. This in fact, this is the question people are trying to answer, and there are two completely different views on this, because the moment you define genomic resources or the DSI as genetic resource, then the rules and regulations that apply for genetic resources for access and benefit sharing will also apply for this. Then whether the international framework or the member countries have wherewithal, do we have facility or do we have capability to implement that, depends upon different countries. For example, if I want to regulate the access of sequence data, can I do that? I cannot, because the sequence data is stored elsewhere. For example, I can request Bart for some accessions of banana. So then he will decide. It is his discretion based on the guidelines that are existing, whether they can be provided to us or not. Now, if the sequences of the same banana accessions are present elsewhere, the gene bank where Bart is working will not have any say on that. So it becomes very difficult on something which is remotely present. So if you define something as genetic resource, that means whether the genomic resource or the sequences are part of genetic resource, it has got very serious implications, which is why people are trying to understand how do we work on that. So as on today, the DSI is not defined as genetic resource.
Okay, thank you very much, Dr. Sunil. Another question from Dr. Yudhistira to Dr. Sunil is about the basmati. We Indonesia, if we commercialize the basmati in Indonesia, what is the consequence for us?
Yeah, a very interesting question, and there can be many other questions. We had recently asked, we had got a similar question from Bangladesh about a similar situation, exactly. Now, please understand, in the definitions, I tried to explain to this, but because of paucity of time, I did not spend too much on that time on that. One is on the germplasm accessions. That means basic germplasm accessions. The other ones are final products which are generated, which are built based on using these germplasm accessions, such as varieties, final varieties. For example, when we access material from CG gene banks, if you ask for germplasm accessions that are different, if you ask for the nurseries that are different, that means the breeding lines, elite lines, if you ask for the varieties which are developed, they are governed differently. Now, if somebody wants to work on a basmati variety, which is released formally from India using the formal procedures and also registered for plant breeders' rights, they are governed differently. You can enter into a bilateral agreement and then start working on that depending upon how your terms and conditions are there. But if you want to access what is called as a land race of basmati, original land races of basmati, then the questions of whatever I mentioned, the governance of access and benefit sharing will come into picture. That also you can contact specific people, which I mentioned that institutions are there. All these things are possible. These are possible only we enter into, let's say, engaging with each other. And I did not tell here that the best way of accessing material is collaborative research. The best way of accessing any material is collaborative research. The moment you enter into collaborative research, it's a win-win situation for everybody. So this is what is my immediate answer to this question. Thank you.
Okay, thank you, Dr. Sunil. Another question is to Dr. Panish. It's come from Dr. Madri Tasma. The question is, can we preserve genetic identity from the changing its genetic identity of genetic material conserved using long-term in vitro culture method? Please, Dr. Panish.
Okay, yes, this is also a good question. When you store material in vitro, there is something which is called somaclonal variation. This means this is the term they use for mutation that can arise during in vitro culture. And this is something that happens more frequently than that it happens in nature, because in vitro material is under stress conditions. You apply different growth regulators, you have these small plants, so it's under stress. So there is a chance that when you store something in vitro for 10 years, like in the case of banana, that you find some abnormalities. But when you apply cryopreservation, in fact, all metabolical, physical, chemical processes are arrested. So in theory, when you store it in liquid nitrogen, there are no changes taking place. So therefore, also cryopreservation is more secure because you stop the changes taking place. So yes, I hope that this answers your question. In liquid nitrogen, when you store something for 1 month or for 100 years, in theory, no changes take place. But in vitro, that's another case.
Okay, thank you very much, Dr. Panish. Another question, Dr. Panish, from Dr. Sri Hartati. The question is how to monitor the viability of genetic material stored using cryopreservation to ensure that it is still good condition, and how frequently should monitoring be performed?
Yes, okay. Also an interesting question that often comes up, of course. So when we do cryopreservation, we preserve, let's say, 100 meristems, and 20 meristems we store in liquid nitrogen for 1 hour, and we regenerate them, and the other 80, they will remain in the liquid nitrogen for long-term conservation. So these 20 meristems we regenerate and see whether we can grow out plants from them. It is, of course, always important after cryopreservation to see whether a full plant can be regenerated, because there are many publications that are reporting survival, but survival is not enough. You need to have a good, healthy plant growing from the stored materials. And this is something, when you read literature, please take into account, or please look carefully, that people talk about plant regeneration and not just survival. Yes, thanks.
Yeah, thank you, Dr. Panis. You still have one question. This is very global, very big question, I think. What is the strategy to obtain high success of plant cryopreservation?
Oh, the strategy. First of all, yeah, you need first to develop an efficient protocol. And such a protocol can really depend from one species to another. So you have to optimize different parameters. Also, even within a species, you have accessions that respond good. You have accessions that respond bad. First of all, you need a good protocol, and then you need to find funds to put the things, a big collection, really into liquid nitrogen. And that is often where such initiatives block. Sometimes you get money to execute a research, to publish a paper, but then finally you need to pay the salaries of the technicians that have to put the material in liquid nitrogen. So one of the strategies is find enough funds to put the things in liquid nitrogen.
Okay.
Yes.
Okay, thank you, Dr. Sunil. Dr. Sunil and Dr. Panis. I think there is no other question, whether directly in the raise hand or in the chat box. Oh, I think still one. Okay, no, I think no question. So I believe that your presentation, Dr. Sunil and Dr. Panis, is very interesting regarding the management of the plant genetic resources, policy, and the governance, and also strategy of the plant preservation. It's very important and very interesting. I believe that there are still many questions, but yeah, maybe they still thinking how to we can contact directly to you. I think that's all our presentation today. Thank you very much, Dr. Sunil. Thank you very much, Dr. Panis. And I give back the time to Miss Anna. Please.
Thank you. Okay, thank you, Bapak Agung Hendriadi, for the session, and to our wonderful speaker for the informative speech. Excellencies, ladies and gentlemen, to honor our special invited speakers and moderator, we will have certificate of appreciation that will be given virtually by the event coordinator of ICFAS 2022, Bapak Arif Arifianto, MSc. So please, I will invite to the screen for our first invited speaker, Dr. Sunil Acak, Bapak Arif Arianto, MSc and the host to share the certificate.
Yes, thank you very much, Anna. Yes, on behalf of the International Conference Committee and Research Organization of Agriculture Food BRIN, I would like to say thank you for Dr. Sunil Archak for your informative presentation and discussion of plant genetic policy and governance. Again, to Dr. Bart Penis, thank you and highly appreciate for your time. Sure, it must be hard for you because now I know in Europe on 5 o'clock in the morning, right? Yes. Okay, and also for your presentation, which is very important to develop the strategy of plant genetic conservation to respond our global climate change. I think this topic is very important for all, yeah, for us. And also, we would like to say thank you to Bapak Dr. Agung Hendriadi for moderating this meeting very well and very interactive. Thank you very much, Dr. Agung.
Thank you, Bapak Arif Arianto, MSc.
Okay, thank you.
Excellencies, ladies and gentlemen, before we are having our lunch break and Friday prayer, please allow me to inform you that the parallel session will begin at 12:40 p.m. and you may rejoin the Zoom at 12:30 and prepare to migrate to the breakout room. And so have a nice afternoon rest and see you at 12:30. Thank you.
National Research and Innovation Agency, a synergy in an orchestra. established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Indonesia, terdapat 14,3 juta hektar lahan perkebunan kelapa sawit. Selama ini, lahan perkebunan tersebut hanya dimanfaatkan untuk pertanaman monokrop. Padahal, produktivitas dan efisiensi lahan dapat ditingkatkan dengan diversifikasi, salah satunya dengan mengintegrasikan perkebunan sawit dengan pembiakan sapi. Lahan perkebunan yang sangat luas berpotensi sebagai sumber pakan hijauan murah untuk penggembalaan sapi. Dari sisi demand, Indonesia sangat membutuhkan pasokan anak sapi atau sapi bakalan untuk digemukkan. Pemerintah tengah menggalakkan pembiakan sapi di dalam negeri untuk mengurangi beban impor. Beberapa perusahaan telah menerapkan integrasi perkebunan sawit dengan pembiakan sapi ini. Misalnya PT Buana Karya Bakti di Kalimantan Selatan dan PT Kalteng Andini Palmal Sari di Kalimantan Tengah. Usaha yang mereka lakukan dinamakan Sistem Integrasi Sapi Kelapa Sawit, disingkat SISKA.
Manajemen kebun ternyata bisa mengintegrasikan sawit dengan sapi, yang kemudian bisa menghasilkan beberapa hal yang kami anggap itu adalah sesuatu yang baik. Suatu saat ini akan bisa menjadi bisnis alternatif sebagai diversifikasi.
Di lahan kelapa sawit tumbuh berbagai macam rumput yang merupakan gulma untuk perkebunan sawit. Namun, sebagian merupakan rumput yang bisa dimanfaatkan sebagai pakan sapi. Sapi digembalakan di bawah sawit, dan mereka bisa merumput dengan bebas. Dalam model SISKA, kebun sawit dibagi dalam beberapa blok penggembalaan. Kelompok-kelompok sapi dibiarkan menggembala dan makan di sebuah blok dan pindah ke blok lainnya keesokan harinya. Perputaran ini disebut sebagai rotasi grazing dan akan berjalan selama 3 bulan. Bagi perkebunan, sapi berperan sebagai biological mower yang membersihkan gulma dan mengurangi biaya pembersihan. Perkebunan pun berkurang ketergantungannya terhadap penggunaan herbisida dan bahan kimia lainnya.
Kita dari kebun dituntut untuk bisa efisien mengenai biaya perawatan. Nah, setelah adanya sapi, sekarang kita sangat terbantu, Pak. Pertama, untuk perawatan kita hanya selektif nih. Jadi untuk gulma-gulma yang terbilang daun lunak, ini tidak kita kendalikan karena nanti bakal jadi makanan sapi. Kita hanya mengendalikan yang sifatnya berbatang keras seperti anakan kayu.
Ini adalah satu bisnis yang sangat efisien. Karena apa? Karena biaya untuk usaha peternakan itu adalah biaya pakan. Nah, ketika pakan itu bisa didapat dari sumber pakan alami yang ada di kebun sawit, yang pasti biayanya akan lebih murah.
Membiarkan sapi di perkebunan sawit tentu bukan hal mudah. Pengelolaan ternak dan perkebunan harus selaras, mengakomodasikan kebutuhan ternak sapi maupun pekerjaan di kebun, dan agar keduanya harmonis. Selain itu, ternak membutuhkan perhatian 24 jam per hari.
Adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
A sustainable food production for our growing world population requires modern breeding strategies and effective quality control procedures for agriculture and aquaculture. This can be achieved through the production of high-quality seeds and improved yield in plant and seafood farming. An important tool for breeding is the determination of ploidy levels. The ploidy level of an organism is characterized by the number of chromosomes pairs. This is specific to each species. Deviations from regular ploidy levels can have positive or negative effects on the individual. An increased number of chromosome sets, called polyploidy, can produce faster growing and bigger plants or seafood, or create plants with an increased production of secondary metabolites like essential oils. To get plants with seedless fruits, plant breeders produce triploid, sterile individuals. Checking the ploidy levels of such plants guarantees that only seedless fruits are formed. Negative effects can be induced by aneuploidy, characterized by an abnormal number of chromosomes. Aneuploid plants are often stunted. This makes checking for aneuploidy important in quality control of seed production. In all these instances, checking ploidy levels helps to improve yield, quality, and productivity. So farmers gain more benefits with the same amount of investment. For more than 35 years, Sysmex Partech has been the leading supplier of complete solutions for ploidy analysis, offering a broad range of dedicated reagents, instruments, and analyzing software optimized for different sample material and organisms like rice, potatoes, sugarcane, corn, and oysters. Even challenging oxidized plant material becomes easy to handle with our solutions. Sysmex's unique complete solution for ploidy analysis with dedicated instruments, reagents, and analysis software like the automated ploidy detection tool support manual and automated high-throughput measures and analysis in accordance with ISO and REACH guidelines. Start now and benefit from reliable, high-quality and cost-efficient solutions based on the Sysmex experience.
ITS Sains Indonesia merupakan salah satu divisi bisnis dari ITS Group yang berkantor utama di Singapura. ITS Sains Indonesia menawarkan berbagai macam instrumen analitik dan medis, peralatan laboratorium umum, bahan kimia, dan bahan habis pakai lainnya. Selama lebih dari 40 tahun, ITS Group bergerak maju dengan visi memberikan pengaruh di dalam industri yang kami jalani. Kultur perusahaan kami untuk selalu mengedepankan profesionalisme, partnership, dan performance. ITS Group tersebar di berbagai negara Asia Tenggara, yaitu Malaysia, Singapura, Thailand, Vietnam, Indonesia, Filipina, dan Kamboja. ITS Sains Indonesia memiliki 2 kantor yang berlokasi di Kelapa Gading, Jakarta Utara, dan Sukomanunggal, Surabaya. Berikut portfolio produk yang kami representasikan di Indonesia. Produk-produk yang kami representasikan merupakan produk-produk unggulan di bidangnya. Selain itu, kami juga bangga bekerja sama dengan berbagai pihak, mulai dari lembaga pemerintah, industri, hingga universitas. Bila Anda memerlukan bantuan untuk laboratorium Anda, hubungi kami. ITS Sains Indonesia, Bridging Science, Inspiring Possibilities.
Wakil Presiden
Presiden Republik Indonesia tiba di tempat acara.
Besar harapan kami agar fasilitas riset pangan BRIN ini pada akhirnya dapat mendukung pengembangan produk halal dan sehat khas Indonesia, terutama produk-produk yang berasal dari bahan lokal dan komoditas pangan tradisional.
Riset dan inovasilah yang mendasari pertumbuhan dunia usaha dan juga menciptakan lapangan kerja yang akan mendorong pertumbuhan ekonomi negara.
Kelompok tani Kapokran Tani Maju yang posisinya ada di Desa Wonorejo, Kecamatan Satui, Kabupaten Tanah Tumbu. Bagi kelompok tani kita bermanfaat sekali, menggunakan biaya operasional baik dari peternak yang biasanya mengembala itu menghabiskan dalam 2 hari itu 3 liter bensin. Manfaatnya itu satu, keduanya dari tingkat keamanan, dari tingkat pemeliharaan kita bisa terkontrol. Mudah-mudahan dengan adanya komunikasi dengan perusahaan kita bisa maju Pak di bidang ternak sapi ini.
Kalau ini kan dijaga baik-baik.
Nama saya Sarip, asisten Abdeling Wonorejo. Dengan adanya sapi di kebun itu salah satunya adalah mengurangi biaya-biaya operasional yang ada di Abdeling. Kita ada koordinasi mengenai rotasi-rotasi grazing, kemudian kita berhubungan dengan rotasi-rotasi panen. Jadi itu yang sebagai patokan kami di Abdeling. Kami pegang ada semacam peta yang dibentuk, yang diolah rotasi dari blok ke blok. Kalau di sini untuk sementara rencananya semua blok Pak. Jadi 1052 hektar.
Semua orang berhak mendapat kesempatan yang lebih baik, dan kami percaya itu. Karenanya, sudah 30 tahun kami dedikasikan diri untuk berjalan denganmu, sahabat kami, para petani dari seluruh pelosok negeri. Sebuah mimpi besar, memang, tetapi kami yakin bisa dimulai dari langkah terkecil. Yang juga paling penting, seperti mengembangkan benih berkualitas hasil penelitian yang menjadi ciri khas kami. Jarak dan medan yang harus ditempuh bukan halangan bagi kami untuk terjun langsung ke lapangan. Kadang penuh keringat, kadang ditemani terik yang menyengat. Tak masalah, karena disinilah perjalanan kami bersamamu dimulai. Percobaan demi percobaan kami lakukan bersama untuk mencari mana varietas terbaik nantinya, yang kami rawat sepenuh hati dalam pengawasan terbaik, agar tiap benihnya mampu menyerap nutrisi bumi sepenuhnya, hingga mereka terus tumbuh sehat sampai hasil panen terlihat. Prosesnya memang panjang dan tak mudah, tetapi dengan inilah benih-benih terpilih sampai ke genggamanmu. Untuk kemudian dikemas menjadi produk cap panah merah yang akan membantu petani-petani selanjutnya. Kami sadar ada banyak hal yang masih harus dilakukan untuk maju bersamamu. Karenanya, pelatihan tercipta untuk kami, supaya bisa selalu siaga membantumu. Dan untukmu, kembangkan kemampuan bertani dengan bantuan ilmu serta teknologi terkini. Kami juga selalu siap menjawab setiap pertanyaanmu. Di setiap pendampingan yang kamu perlukan, kami ada untukmu. Dan ketika kami melihatmu berhasil, mendapat kesempatan, serta mencapai apa yang kamu mimpikan, itulah pencapaian terbesar kami dalam menjadi sahabat yang paling baik bagimu. Terima kasih telah berjalan bersama kami selama 30 tahun ini. Mari terus menjadi sahabat dalam berpuluh tahun mendatang. ICFAS Indonesia, sahabat petani yang paling baik.
ITS Sains Indonesia merupakan salah satu divisi bisnis dari ITS Group yang berkantor utama di Singapura. ITS Sains Indonesia menawarkan berbagai macam instrumen analitik dan medis, peralatan laboratorium umum, bahan kimia, dan bahan habis pakai lainnya. Selama lebih dari 40 tahun, ITS Group bergerak maju dengan visi memberikan pengaruh di dalam industri yang kami jalani. Kultur perusahaan kami untuk selalu mengedepankan profesionalisme, partnership, dan performance. ITS Group tersebar di berbagai negara Asia Tenggara, yaitu Malaysia, Singapura, Thailand, Vietnam, Indonesia, Filipina, dan Kamboja. ITS Sains Indonesia memiliki 2 kantor yang berlokasi di Kelapa Gading, Jakarta Utara, dan Sukomanunggal, Surabaya. Berikut portfolio produk yang kami representasikan di Indonesia. Produk-produk yang kami representasikan merupakan produk-produk unggulan di bidangnya.
Apa lagi? Apa?
Selain itu, kami juga bangga bekerja sama dengan berbagai pihak, mulai dari lembaga pemerintah, industri, hingga universitas. Bila Anda memerlukan bantuan untuk laboratorium Anda, hubungi kami. ITS Science Indonesia, Bridging Science, Inspiring Possibilities.
Pertanian adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Saya kira kedepannya sangat baik prospeknya di kedua sisi ya, baik dalam peningkatan sawit itu sendiri maupun peningkatan produktivitas di sapinya, pemenuhan daging nasional.
Dan kami harapkan bahwa SSP ini bisa mendampingi sampai dengan tahun 2026 atau bahkan seterusnya selama pengembangan Siska dilaksanakan di Provinsi Kalimantan Selatan.
Dengan mengucapkan Bismillahirrahmanirrahim, fasilitas riset pangan di Pusat Riset Teknologi dan Proses Pangan, Badan Riset dan Inovasi Nasional sebagai laboratorium rujukan riset halal Indonesia, saya nyatakan diresmikan.
adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
Banyak banget pengalaman baru dan ilmu baru yang saya dapat. Pengalaman di sini sangat bermanfaat dan benar-benar berharga bagi saya. Selama saya magang di Siska,
tentunya saya belajar banyak hal, tidak hanya secara teori,
tapi juga praktis secara langsung di lapang, terutama mengenai integrasi sawit sapi dan juga manajemen pastura.
Harapan saya bisa belajar lebih lanjut di Australia.
MBKM PT Siska, always be the best.
Yes, yes, yes.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of good manufacturing process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies. National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. to enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Indonesia, terdapat 14,3 juta hektar lahan perkebunan kelapa sawit. Selama ini, lahan perkebunan tersebut hanya dimanfaatkan untuk pertanaman monokrop. Padahal, produktivitas dan efisiensi lahan dapat ditingkatkan dengan diversifikasi, salah satunya dengan mengintegrasikan perkebunan sawit dengan pembiakan sapi. Lahan perkebunan yang sangat luas berpotensi sebagai sumber pakan hijauan murah untuk penggembalaan sapi. Dari sisi demand, Indonesia sangat membutuhkan pasokan anak sapi atau sapi bakalan untuk digemukkan. Pemerintah tengah menggalakkan pembiakan sapi di dalam negeri untuk mengurangi beban impor. Beberapa perusahaan telah menerapkan integrasi perkebunan sawit dengan pembiakan sapi ini. Misalnya PT Buana Karya Bakti di Kalimantan Selatan dan PT Kalteng Andini Palmal Sari di Kalimantan Tengah. Usaha yang mereka lakukan dinamakan Sistem Integrasi Sapi Kelapa Sawit, disingkat SISKA.
Manajemen kebun ternyata bisa mengintegrasikan sawit dengan sapi, yang kemudian bisa menghasilkan beberapa hal yang kami anggap itu adalah sesuatu yang baik. Suatu saat ini akan bisa menjadi bisnis alternatif sebagai diversifikasi.
Di lahan kelapa sawit tumbuh berbagai macam rumput yang merupakan gulma untuk perkebunan sawit. Namun, sebagian merupakan rumput yang bisa dimanfaatkan sebagai pakan sapi. Sapi digembalakan di bawah sawit, dan mereka bisa merumput dengan bebas. Dalam model SISKA, kebun sawit dibagi dalam beberapa blok penggembalaan. Kelompok-kelompok sapi dibiarkan menggembala dan makan di sebuah blok dan pindah ke blok lainnya keesokan harinya. Perputaran ini disebut sebagai rotasi grazing dan akan berjalan selama 3 bulan. Bagi perkebunan, sapi berperan sebagai biological mower yang membersihkan gulma dan mengurangi biaya pembersihan. Perkebunan pun berkurang ketergantungannya terhadap penggunaan herbisida dan bahan kimia lainnya.
Kita dari kebun dituntut untuk bisa efisien mengenai biaya perawatan. Nah, setelah adanya sapi, sekarang kita sangat terbantu, Pak. Pertama, untuk perawatan kita hanya selektif nih. Jadi, untuk gulma-gulma yang terbilang daun lunak, ini tidak kita kendalikan karena nanti bakal jadi makanan sapi. Kita hanya mengendalikan yang sifatnya berbatang keras seperti anakan kayu.
Ini adalah satu bisnis yang sangat efisien. Karena apa? Karena biaya untuk usaha peternakan itu adalah biaya pakan. Nah, ketika pakan itu bisa didapat dari sumber pakan alami yang ada di kebun sawit, yang pasti biayanya akan lebih murah.
Membiarkan sapi di perkebunan sawit tentu bukan hal mudah. Pengelolaan ternak dan perkebunan harus selaras, mengakomodasikan kebutuhan ternak sapi maupun pekerjaan di kebun, dan agar keduanya harmonis. Selain itu, ternak membutuhkan perhatian 24 jam per hari.
Adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
A sustainable food production for our growing world population requires modern breeding strategies and effective quality control procedures for agriculture and aquaculture. This can be achieved through the production of high-quality seeds and improved yield in plant and seafood farming. An important tool for breeding is the determination of ploidy levels. The ploidy level of an organism is characterized by the number of chromosomes pairs. This is specific to each species. Deviations from regular ploidy levels can have positive or negative effects on the individual. An increased number of chromosome sets, called polyploidy, can produce faster growing and bigger plants or seafood, or create plants with an increased production of secondary metabolites like essential oils. To get plants with seedless fruits, plant breeders produce triploid, sterile individuals. Checking the ploidy levels of such plants guarantees that only seedless fruits are formed. Negative effects can be induced by aneuploidy, characterized by an abnormal number of chromosomes. Aneuploid plants are often stunted. This makes checking for aneuploidy important in quality control of seed production. In all these instances, checking ploidy levels helps to improve yield quality, and productivity. So farmers gain more benefits with the same amount of investment. For more than 35 years, Sysmex Partech has been the leading supplier of complete solutions for ploidy analysis, offering a broad range of dedicated reagents, instruments, and analyzing software optimized for different sample material and organisms like rice, potatoes, sugarcane, corn, and oysters. Even challenging oxidized plant material becomes easy to handle with our solutions. Sysmex's unique complete solution for ploidy analysis with dedicated instruments, reagents, and analysis software like the automated ploidy detection tool support manual and automated high-throughput measures and analysis in accordance with ISO and REACH guidelines. Start now and benefit from reliable, high-quality and cost-efficient solutions based on the Sysmex experience.
ITS Sains Indonesia merupakan salah satu divisi bisnis dari ITS Group yang berkantor utama di Singapura. ITS Sains Indonesia menawarkan berbagai macam instrumen analitik dan medis, peralatan laboratorium umum, bahan kimia, dan bahan habis pakai lainnya. Selama lebih dari 40 tahun, ITS Group bergerak maju dengan visi memberikan pengaruh di dalam industri yang kami jalani. Kultur perusahaan kami untuk selalu mengedepankan profesionalisme, partnership, dan performance. ITS Group tersebar di berbagai negara Asia Tenggara, yaitu Malaysia, Singapura, Thailand, Vietnam, Indonesia, Filipina, dan Kamboja. ITS Sains Indonesia memiliki 2 kantor yang berlokasi di Kelapa Gading, Jakarta Utara, dan Sukomanunggal, Surabaya. Berikut portfolio produk yang kami representasikan di Indonesia. Produk-produk yang kami representasikan merupakan produk-produk unggulan di bidangnya. Selain itu, kami juga bangga bekerja sama dengan berbagai pihak, mulai dari lembaga pemerintah, industri, hingga universitas. Bila Anda memerlukan bantuan untuk laboratorium Anda, hubungi kami. ITS Sains Indonesia, Bridging Science.
Bu Mimi udah ya, Bu? Iya, Dek, tolong kunciin dulu, Dek.
Ya, kelihatannya udah mulai ini. Cuma, oke. Ya.
Okay. Excellencies, ladies and gentlemen, welcome back to the first International Conference on Food and Agricultural Sciences, ICFAS 2022. Okay.
Kenapa? Udah ngomong MC-nya?
Breakout room.
Thank you. Excellencies, ladies and gentlemen. Welcome back to the first International Conference on Food and Agricultural Sciences, ICFAS 2022, with the theme Advanced Agricultural Technology to deal with climate change issue for achieving food security. Before we enter the parallel session, please allow me to convey some information. Number one, we will have the parallel session in 15 breakout rooms with 10 presenters for each breakout room. And you may enter the breakout room according to the field of study by clicking the breakout room button below and choose your destination breakout room according to the list from the cabinet. Number two, each participant should present their paper within 10 minutes and 5 minutes to discussion as mentioned in the program book as well. And number three, we will back to the main room at 3:25 p.m. after the parallel session. And the last, the following agenda is the announcement of the best presenter, the best paper, and the best participant, which will be announced by the chairman of ICFAS 2022. So thank you, have a great conference, and see you at 3:25 p.m. in the main room.
Mbak Ana, berkerumnya belum muncul. Berkerumnya belum muncul, Mbak.
Okay, well, waiting for the breakout room, you can see the rules how to join the breakout room. Please enjoy, and please the host to share the rules of the breakout room.
Dear participants, please attention. Please rename your name on your video as the required from the program book. Thank you.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance
Dear participants in the main room, if you want to join to the room, please rename your video name as like R1 if you want to join to the room 1. So for example, before, as my name Rismayani, before Rismayani, I would like to join in the one room, so I write R1. Thank you.
Center for Technology Services and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies. National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
A sustainable food production for our growing world population requires modern breeding strategies and effective quality control procedures for agriculture and aquaculture. This can be achieved through the production of high-quality seeds and improved yield in plant and seafood farming. An important tool for breeding is the determination of ploidy levels. The ploidy level of an organism is characterized by the number of chromosomes pairs. This is specific to each species. Deviations from regular ploidy levels can have positive or negative effects on the individual. An increased number of chromosome sets, called polyploidy, can produce faster growing and bigger plants or seafood, or create plants with an increased production of secondary metabolites like essential oils. To get plants with seedless fruits, plant breeders produce triploid, sterile individuals. Checking the ploidy levels of such plants guarantees that only seedless fruits are formed. Negative effects can be induced by aneuploidy, characterized by an abnormal number of chromosomes. Aneuploid plants are often stunted. This makes checking for aneuploidy important in quality control of seed production. In all these instances, checking ploidy levels helps to improve yield, quality, and productivity. So farmers gain more benefits with the same amount of investment. For more than 35 years, Sysmex Partec has been the leading supplier of complete solutions for ploidy analysis, offering a broad range of dedicated reagents, instruments, and analyzing software optimized for different sample material and organisms like rice, potatoes, sugarcane, corn, and oysters. Even challenging oxidized plant material becomes easy to handle with our solutions. Sysmex's unique complete solution for ploidy analysis with dedicated instruments, reagents, and analysis software like the automated ploidy detection tool support manual and automated high-throughput measures and analysis in accordance with ISO and REACH guidelines. Start now and benefit from reliable, high-quality and cost-efficient solutions based on the Sysmex experience.
Indonesia terdapat 14,3 juta hektar lahan perkebunan kelapa sawit. Selama ini lahan perkebunan tersebut hanya dimanfaatkan untuk pertanaman monokrop. Padahal produktivitas dan efisiensi lahan dapat ditingkatkan dengan diversifikasi, salah satunya dengan mengintegrasikan perkebunan sawit dengan pembiakan sapi. Lahan perkebunan yang sangat luas berpotensi sebagai sumber pakan hijauan murah untuk penggembalaan sapi. Dari sisi demand, Indonesia sangat membutuhkan pasokan anak sapi atau sapi bakalan untuk digemukkan. Pemerintah tengah menggalakkan pembiakan sapi di dalam negeri untuk mengurangi beban impor. Beberapa perusahaan telah menerapkan integrasi perkebunan sawit dengan pembiakan sapi ini. Misalnya PT Buana Karya Bakti di Kalimantan Selatan dan PT Kalteng Andini Palmal Sari di Kalimantan Tengah. Usaha yang mereka lakukan dinamakan Sistem Integrasi Sapi Kelapa Sawit, disingkat SISKA.
Manajemen kebun ternyata bisa mengintegrasikan sawit dengan sapi yang kemudian bisa menghasilkan beberapa hal yang kami anggap itu adalah sesuatu yang baik. Suatu saat ini akan bisa menjadi bisnis alternatif sebagai diversifikasi.
Di lahan kelapa sawit tumbuh berbagai macam rumput yang merupakan gulma untuk perkebunan sawit. Namun, sebagian merupakan rumput yang bisa dimanfaatkan sebagai pakan sapi. Sapi digembalakan di bawah sawit dan mereka bisa merumput dengan bebas. Dalam model SISKA, kebun sawit dibagi dalam beberapa blok penggembalaan. Kelompok-kelompok sapi dibiarkan menggembala dan makan di sebuah blok dan pindah ke blok lainnya keesokan harinya. Perputaran ini disebut sebagai rotasi grazing dan akan berjalan selama 3 bulan. Bagi perkebunan, sapi berperan sebagai biological mower yang membersihkan gulma dan mengurangi biaya pembersihan. Perkebunan pun berkurang ketergantungannya terhadap penggunaan herbisida dan bahan kimia lainnya.
Kita dari kebun dituntut untuk bisa efisien mengenai biaya perawatan. Nah, setelah adanya sapi, sekarang kita sangat terbantu, Pak. Pertama, untuk perawatan kita hanya selektif nih. Jadi, untuk gulma-gulma yang terbilang daun lunak, ini tidak kita kendalikan karena nanti bakal jadi makanan sapi. Kita hanya mengendalikan yang sifatnya berbatang keras seperti anakan kayu.
Ini adalah satu bisnis yang sangat efisien. Karena apa? Karena biaya untuk usaha peternakan itu adalah biaya pakan. Nah, ketika pakan itu bisa didapat dari sumber pakan alami yang ada di kebun sawit, yang pasti biayanya akan lebih murah.
Membiarkan sapi di perkebunan sawit tentu bukan hal mudah. Pengelolaan ternak dan perkebunan harus selaras, mengakomodasikan kebutuhan ternak sapi maupun pekerjaan di kebun, dan agar keduanya harmonis. Selain itu, ternak membutuhkan perhatian 24 jam per hari.
Adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada Crop Life Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. Crop Life Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
ITS Sains Indonesia merupakan salah satu divisi bisnis dari ITS Group yang berkantor utama di Singapura. ITS Sains Indonesia menawarkan berbagai macam instrumen analitik dan medis, peralatan laboratorium umum, bahan kimia, dan bahan habis pakai lainnya. Selama lebih dari 40 tahun, ITS Group bergerak maju dengan visi memberikan pengaruh di dalam industri yang kami jalani. Kultur perusahaan kami untuk selalu mengedepankan profesionalisme, partnership, dan performance. ITS Group tersebar di berbagai negara Asia Tenggara, yaitu Malaysia, Singapura, Thailand, Vietnam, Indonesia, Filipina, dan Kamboja. ITS Sains Indonesia memiliki 2 kantor yang berlokasi di Kelapa Gading, Jakarta Utara, dan Sukomanunggal, Surabaya. Berikut portfolio produk yang kami representasikan di Indonesia. Produk-produk yang kami representasikan merupakan produk-produk unggulan di bidangnya. Selain itu, kami juga bangga bekerja sama dengan berbagai pihak, mulai dari lembaga pemerintah, industri, hingga universitas. Bila Anda memerlukan bantuan untuk laboratorium Anda, hubungi kami. ITS Sains Indonesia, Bridging Science Inspiring Possibilities.
Wakil Presiden Republik Indonesia,
tiba di tempat acara.
Besar harapan kami agar fasilitas riset pangan BRIN ini pada akhirnya dapat mendukung pengembangan produk halal dan sehat khas Indonesia, terutama produk-produk yang berasal dari bahan lokal dan komoditas pangan tradisional.
Riset dan inovasi lah yang mendasari pertumbuhan dunia usaha dan juga menciptakan lapangan kerja yang akan mendorong pertumbuhan ekonomi negara.
Saya Sutino, kelompok tani Kapokran Tani Maju yang posisinya ada di Desa Wonorejo, Kecamatan Satui, Kabupaten Tanah Sumbung. Bagi kelompok tani kita bermanfaat sekali, menggunakan biaya operasional baik dari peternak yang biasanya mengembala itu menghabiskan dalam 2 hari itu 3 liter bensin. Manfaatnya itu satu, keduanya dari tingkat keamanan, dari tingkat pemeliharaan kita bisa terkontrol. Mudah-mudahan dengan adanya komunikasi dengan perusahaan kita bisa maju Pak di bidang ternak sapi ini.
Kalau ini kan dijaga baik-baik.
Nama saya Sarip, asisten Abdeling Wonorejo. Dengan adanya sapi di kebun itu salah satunya adalah mengurangi biaya-biaya operasional yang ada di Abdeling. Kita ada koordinasi mengenai rotasi-rotasi grazing, kemudian kita berhubungan dengan rotasi-rotasi panen. Jadi itu yang sebagai patokan kami di Abdeling. Kami pegang ada semacam peta yang dibentuk, yang diolah rotasi dari blok ke blok. Kalau di sini untuk sementara rencananya semua blok Pak. Jadi 1052 hektar.
Semua orang berhak mendapat kesempatan yang lebih baik, dan kami percaya itu. Karenanya, sudah 30 tahun kami dedikasikan diri untuk berjalan denganmu, sahabat kami, para petani dari seluruh pelosok negeri. Sebuah mimpi besar, memang, tetapi kami yakin bisa dimulai dari langkah terkecil. Yang juga paling penting, seperti mengembangkan benih berkualitas hasil penelitian yang menjadi ciri khas kami. Jarak dan medan yang harus ditempuh bukan halangan bagi kami untuk terjun langsung ke lapangan. Kadang penuh keringat kadang ditemani terik yang menyengat. Tak masalah, karena disinilah perjalanan kami bersamamu dimulai. Percobaan demi percobaan kami lakukan bersama untuk mencari mana varietas terbaik nantinya, yang kami rawat sepenuh hati dalam pengawasan terbaik, agar tiap benihnya mampu menyerap nutrisi bumi sepenuhnya, hingga mereka terus tumbuh sehat sampai hasil panen terlihat. Prosesnya memang panjang dan tak mudah, tetapi dengan inilah benih-benih terpilih sampai ke genggamanmu. Untuk kemudian dikemas menjadi produk cap panah merah yang akan membantu petani-petani selanjutnya. Kami sadar ada banyak hal yang masih harus dilakukan untuk maju bersamamu. Karenanya, pelatihan tercipta untuk kami, supaya bisa selalu siaga membantumu. Dan untukmu, kembangkan kemampuan bertani dengan bantuan ilmu serta teknologi terkini. Kami juga selalu siap menjawab setiap pertanyaanmu. Di setiap pendampingan yang kamu perlukan, kami ada untukmu. Dan ketika kami melihatmu berhasil, mendapat kesempatan, serta mencapai apa yang kamu mimpikan, itulah pencapaian terbesar kami dalam menjadi sahabat yang paling baik bagimu. Terima kasih telah berjalan bersama kami selama 30 tahun ini. Mari terus menjadi sahabat dalam berpuluh tahun mendatang. ICFAS Indonesia, sahabat petani yang paling baik.
ITS Sains Indonesia merupakan salah satu divisi bisnis dari ITS Group yang berkantor utama di Singapura. ITS Sains Indonesia menawarkan berbagai macam instrumen analitik dan medis, peralatan laboratorium umum, bahan kimia, dan bahan habis pakai lainnya. Selama lebih dari 40 tahun, ITS Group bergerak maju dengan visi memberikan pengaruh di dalam industri yang kami jalani. Kultur perusahaan kami untuk selalu mengedepankan profesionalisme, partnership, dan performance. ITS Group tersebar di berbagai negara Asia Tenggara, yaitu Malaysia, Singapura, Thailand, Vietnam, Indonesia, Filipina, dan Kamboja. ITS Sains Indonesia memiliki 2 kantor yang berlokasi di Kelapa Gading, Jakarta Utara, dan Sukomanunggal, Surabaya. Berikut portfolio produk yang kami representasikan di Indonesia. Produk-produk yang kami representasikan merupakan produk-produk unggulan di bidangnya. Selain itu, kami juga bangga bekerja sama dengan berbagai pihak, mulai dari lembaga pemerintah, industri, hingga universitas. Bila Anda memerlukan bantuan untuk laboratorium Anda, hubungi kami. ITS Science Indonesia, Bridging Science, Inspiring Possibilities.
Pertanian adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. Life Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal, yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Saya kira kedepannya sangat baik prospeknya di kedua sisi ya, baik dalam peningkatan sawit itu sendiri maupun peningkatan produktivitas di sapinya, pemenuhan daging nasional.
Dan kami harapkan bahwa SSP ini bisa mendampingi sampai dengan tahun 2026 atau bahkan seterusnya selama pengembangan Siska dilaksanakan di Provinsi Kalimantan Selatan.
Dengan mengucapkan Bismillahirrahmanirrahim, fasilitas riset pangan di Pusat Riset Teknologi dan Proses Pangan, Badan Riset dan Inovasi Nasional sebagai laboratorium rujukan riset halal Indonesia, saya nyatakan diresmikan.
adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
Banyak banget pengalaman baru dan ilmu baru yang saya dapat. Pengalaman di sini sangat bermanfaat dan benar-benar berharga bagi saya. Selama saya magang di Siska,
tentunya saya belajar banyak hal, tidak hanya secara teori,
tapi juga praktis secara langsung di lapang, terutama mengenai integrasi sawit sapi dan juga manajemen pastura.
Harapan saya bisa belajar lebih lanjut di Australia.
MBKM PT Siska, always be the best. Yes, yes, yes.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Wakil Presiden Republik Indonesia tiba di tempat acara.
Besar harapan kami agar fasilitas riset pangan BRIN ini pada akhirnya dapat mendukung pengembangan produk halal dan sehat khas Indonesia, terutama produk-produk yang berasal dari bahan lokal dan komoditas pangan tradisional.
Riset dan inovasilah yang mendasari pertumbuhan dunia usaha dan juga menciptakan lapangan kerja yang akan mendorong pertumbuhan ekonomi negara.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Indonesia, terdapat 14,3 juta hektar lahan perkebunan kelapa sawit. Selama ini, lahan perkebunan tersebut hanya dimanfaatkan untuk pertanaman monokrop. Padahal, produktivitas dan efisiensi lahan dapat ditingkatkan dengan diversifikasi, salah satunya dengan mengintegrasikan perkebunan sawit dengan pembiakan sapi. Lahan perkebunan yang sangat luas berpotensi sebagai sumber pakan hijauan murah untuk penggembalaan sapi. Dari sisi demand, Indonesia sangat membutuhkan pasokan anak sapi atau sapi bakalan untuk digemukkan. Pemerintah tengah menggalakkan pembiakan sapi di dalam negeri untuk mengurangi beban impor. Beberapa perusahaan telah menerapkan integrasi perkebunan sawit dengan pembiakan sapi ini. Misalnya PT Buana Karya Bakti di Kalimantan Selatan dan PT Kalteng Andini Palma Lestari di Kalimantan Tengah. Usaha yang mereka lakukan dinamakan Sistem Integrasi Sapi Kelapa Sawit, disingkat SISKA.
Manajemen kebun ternyata bisa mengintegrasikan sawit dengan sapi, yang kemudian bisa menghasilkan beberapa hal yang kami anggap itu adalah sesuatu yang baik. Suatu saat ini akan bisa menjadi bisnis alternatif sebagai diversifikasi.
Di lahan kelapa sawit tumbuh berbagai macam rumput yang merupakan gulma untuk perkebunan sawit. Namun, sebagian merupakan rumput yang bisa dimanfaatkan sebagai pakan sapi. Sapi digembalakan di bawah sawit, dan mereka bisa merumput dengan bebas. Dalam model SISKA, kebun sawit dibagi dalam beberapa blok penggembalaan. Kelompok-kelompok sapi dibiarkan menggembala dan makan di sebuah blok dan pindah ke blok lainnya keesokan harinya. Perputaran ini disebut sebagai rotasi grazing dan akan berjalan selama 3 bulan. Bagi perkebunan, sapi berperan sebagai biological mower yang membersihkan gulma dan mengurangi biaya pembersihan. Perkebunan pun berkurang ketergantungannya terhadap penggunaan herbisida dan bahan kimia lainnya.
Kita dari kebun dituntut untuk bisa efisien mengenai biaya perawatan. Nah, setelah adanya sapi, sekarang kita sangat terbantu, Pak. Pertama, untuk perawatan kita hanya selektif nih. Jadi, untuk gulma-gulma yang terbilang daun lunak, ini tidak kita kendalikan karena nanti bakal jadi makanan sapi. Kita hanya mengendalikan yang sifatnya berbatang keras seperti anakan kayu.
Ini adalah satu bisnis yang sangat efisien. Karena apa? Karena biaya untuk usaha peternakan itu adalah biaya pakan. Nah, ketika pakan itu bisa didapat dari sumber pakan alami yang ada di kebun sawit, yang pasti biayanya akan lebih murah.
Membiarkan sapi di perkebunan sawit tentu bukan hal mudah. Pengelolaan ternak dan perkebunan harus selaras, mengakomodasikan kebutuhan ternak sapi maupun pekerjaan di kebun, dan agar keduanya harmonis. Selain itu, ternak membutuhkan perhatian 24 jam per hari.
Pertanian adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat. Dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
A sustainable food production for our growing world population requires modern breeding strategies and effective quality control procedures for agriculture and aquaculture. This can be achieved through the production of high-quality seeds and improved yield in plant and seafood farming. An important tool for breeding is the determination of ploidy levels. The ploidy level of an organism is characterized by the number of chromosomes pairs. This is specific to each species. Deviations from regular ploidy levels can have positive or negative effects on the individual. An increased number of chromosome sets, called polyploidy, can produce faster growing and bigger plants or seafood, or create plants with an increased production of secondary metabolites like essential oils. To get plants with seedless fruits, plant breeders produce triploid, sterile individuals. Checking the ploidy levels of such plants guarantees that only seedless fruits are formed. Negative effects can be induced by aneuploidy, characterized by an abnormal number of chromosomes. Aneuploid plants are often stunted. This makes checking for aneuploidy important in quality control of seed production. In all these instances, checking ploidy levels helps to improve yield, quality, and productivity. So farmers gain more benefits with the same amount of investment. For more than 35 years, Sysmex Partech has been the leading supplier of complete solutions for ploidy analysis, offering a broad range of dedicated reagents, instruments, and analyzing software optimized for different sample material and organisms like rice, potatoes, sugarcane, corn, and oysters. Even challenging oxidized plant material becomes easy to handle with our solutions. Sysmex's unique complete solution for ploidy analysis with dedicated instruments, reagents, and analysis software like the automated ploidy detection tool, support manual and automated high-throughput measures and analysis in accordance with ISO and REACH guidelines. Start now and benefit from reliable, high-quality and cost-efficient solutions based on the Sysmex experience.
Waduh.
ITS Science Indonesia merupakan salah satu divisi bisnis dari ITS Group yang berkantor utama di Singapura. ITS Science Indonesia menawarkan berbagai macam instrumen analitik dan medis, peralatan laboratorium umum, bahan kimia, dan bahan habis pakai lainnya. Selama lebih dari 40 tahun, ITS Group bergerak maju dengan visi memberikan pengaruh di dalam industri yang kami jalani. Kultur perusahaan kami untuk selalu mengedepankan profesionalisme, partnership, dan performance. ITS Group tersebar di berbagai negara Asia Tenggara, yaitu Malaysia, Singapura, Thailand, Vietnam, Indonesia, Filipina, dan Kamboja. ITS Sains Indonesia memiliki 2 kantor yang berlokasi di Kelapa Gading, Jakarta Utara, dan Sukomanunggal, Surabaya. Berikut portfolio produk yang kami representasikan di Indonesia. Produk-produk yang kami representasikan merupakan produk-produk unggulan di bidangnya. Selain itu, kami juga bangga bekerja sama dengan berbagai pihak, mulai dari lembaga pemerintah, industri, hingga universitas. Bila Anda memerlukan bantuan untuk laboratorium Anda, hubungi kami. ITS Sains Indonesia, Bridging Science Inspiring Possibilities.
Biar saya nggak ada tonggih orang balik sini ya. Bisa kadang isar duluan. Tapi ini 15 di
Republik Indonesia tiba di tempat
besar harapan kami agar fasilitas riset pangan BRIN ini pada akhirnya dapat mendukung pengembangan produk halal dan sehat khas Indonesia, terutama produk-produk yang berasal dari bahan lokal dan komoditas pangan tradisional.
Riset dan inovasi lah yang mendasari pertumbuhan dunia usaha dan juga menciptakan lapangan kerja yang akan mendorong pertumbuhan ekonomi negara.
Saya Sutino, kelompok tani Kapokran Tani Maju yang posisinya ada di Desa Wonorejo, Kecamatan Satui, Kabupaten Tanah Sumbung. Bagi kelompok tani kita bermanfaat sekali, menekan biaya operasional, baik dari peternak yang biasanya mengembala itu menghabiskan dalam 2 hari itu 3 liter bensin. Manfaatnya itu satu, keduanya dari tingkat keamanan, dari tingkat pemeliharaan kita bisa terkontrol. Mudah-mudahan dengan adanya komunikasi dengan perusahaan kita bisa maju Pak di bidang ternak sapi ini.
Kalau ini kan dijaga baik-baik,
Nama saya Sarip, asisten Abdeling Wonorejo. Dengan adanya sapi di kebun itu, salah satunya adalah mengurangi biaya-biaya operasional yang ada di Abdeling. Kita ada koordinasi mengenai rotasi-rotasi grazing, kemudian kita berhubungan dengan rotasi-rotasi panen. Jadi itu yang sebagai patokan kami di Abdeling. Kami pegang ada semacam peta yang dibentuk, yang diolah rotasi dari blok ke blok. Kalau di sini untuk sementara rencananya semua blok, Pak. Jadi 1052 hektar.
Semua orang berhak mendapat kesempatan yang lebih baik, dan kami percaya itu. Karenanya, sudah 30 tahun kami dedikasikan diri untuk berjalan denganmu, sahabat kami, para petani dari seluruh pelosok negeri. Sebuah mimpi besar, tetapi kami yakin bisa dimulai dari langkah yang juga paling penting, seperti mengembangkan benih berkualitas hasil penelitian yang menjadi ciri khas kami. Jarak dan medan yang harus ditempuh bukan halangan bagi kami untuk terjun langsung ke lapangan. Kadang penuh keringat, kadang ditemani terik yang menyengat. Tak masalah, karena disinilah perjalanan kami bersamamu dimulai. Percobaan demi percobaan kami lakukan bersama untuk mencari mana varietas terbaik nantinya, yang kami rawat sepenuh hati dalam pengawasan terbaik, agar tiap benihnya mampu menyerap nutrisi bumi sepenuhnya, hingga mereka terus tumbuh sehat, sampai hasil panen terlihat. Prosesnya memang panjang dan tak mudah. Tetapi dengan inilah, benih-benih terpilih sampai ke genggamanmu. Untuk kemudian dikemas menjadi produk cap panah merah yang akan membantu petani-petani selanjutnya. Kami sadar ada banyak hal yang masih harus dilakukan untuk maju bersamamu. Karenanya, pelatihan tercipta untuk kami, supaya bisa selalu siaga membantumu. Dan untukmu, kembangkan kemampuan bertani dengan bantuan ilmu serta teknologi terkini. Kami juga selalu siap menjawab setiap pertanyaanmu. Di setiap pendampingan yang kamu perlukan, kami ada untukmu. Dan ketika kami melihatmu berhasil, mendapat kesempatan, serta mencapai apa yang kamu mimpikan, itulah pencapaian terbesar kami dalam menjadi sahabat yang paling baik bagimu. Terima kasih telah berjalan bersama kami selama 30 tahun ini. Mari terus menjadi sahabat dalam berpuluh tahun mendatang. ICFAS Indonesia, sahabat petani yang paling baik.
ITS Sains Indonesia merupakan salah satu divisi bisnis dari ITS Group yang berkantor utama di Singapura. ITS Sains Indonesia menawarkan berbagai macam instrumen analitik dan medis, peralatan laboratorium umum, bahan kimia, dan bahan habis pakai lainnya. Selama lebih dari 40 tahun, ITS Group bergerak maju dengan visi memberikan pengaruh di dalam industri yang kami jalani. Kultur perusahaan kami untuk selalu mengedepankan profesionalisme, partnership, dan performance. ITS Group tersebar di berbagai negara Asia Tenggara, yaitu Malaysia, Singapura, Thailand, Vietnam, Indonesia, Filipina, dan Kamboja. ITS Sains Indonesia memiliki 2 kantor yang berlokasi di Kelapa Gading, Jakarta Utara, dan Sukomanunggal, Surabaya. Berikut portfolio produk yang kami representasikan di Indonesia. Produk-produk yang kami representasikan merupakan produk-produk unggulan di bidangnya. Selain itu, kami juga bangga bekerja sama dengan berbagai pihak, mulai dari lembaga pemerintah, industri, hingga universitas. Bila Anda memerlukan bantuan untuk laboratorium Anda, hubungi kami. ITS Sains Indonesia, Bridging Science, Inspiring Possibilities.
Pertanian adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat. Dunia, termasuk Indonesia, dihadapkan pada ancaman kekurangan pangan. berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. through the three directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Saya kira kedepannya sangat baik prospeknya di kedua sisi ya, baik dalam peningkatan sawit itu sendiri maupun peningkatan produktivitas di sapinya, pemenuhan daging nasional.
Dan kami harapkan bahwa SSP ini bisa mendampingi sampai dengan tahun 2026 atau bahkan seterusnya selama pengembangan Siska dilaksanakan di Provinsi Kalimantan Selatan.
Pertanian adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, Penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi standar, serta penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
Banyak banget pengalaman baru dan ilmu baru yang saya dapat.
Pengalaman di sini sangat bermanfaat dan benar-benar berharga bagi saya. Selama saya magang di Siska,
tentunya saya belajar banyak hal, tidak hanya secara teori,
tapi juga praktis secara langsung di lapang,
terutama mengenai integrasi sawit sapi dan juga manajemen pastura.
Harapan saya bisa belajar lebih lanjut di
Australia. MBKM PT Siska, always be the best. Yes, yes, yes.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies. National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the three directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Semua orang berhak mendapat kesempatan yang lebih baik, dan kami percaya itu. Karenanya, sudah 30 tahun kami dedikasikan diri untuk berjalan denganmu, sahabat kami, para petani dari seluruh pelosok negeri. Sebuah mimpi besar, memang, tetapi kami yakin bisa dimulai dari langkah terkecil. Yang juga paling penting, seperti mengembangkan benih berkualitas hasil penelitian yang menjadi ciri khas kami. Jarak dan medan yang harus ditempuh bukan halangan bagi kami untuk terjun langsung ke lapangan. Kadang penuh keringat, kadang ditemani terik yang menyengat. Tak masalah, karena disinilah perjalanan kami bersamamu dimulai. Percobaan demi percobaan kami lakukan bersama untuk mencari mana varietas terbaik nantinya, yang kami rawat sepenuh hati dalam pengawasan terbaik, agar tiap benihnya mampu menyerap nutrisi bumi sepenuhnya, hingga mereka terus tumbuh sehat, sampai hasil panen terlihat. Prosesnya memang panjang dan tak mudah. Tetapi dengan inilah, benih-benih terpilih sampai ke genggamanmu. Untuk kemudian dikemas menjadi produk cap panah merah yang akan membantu petani-petani selanjutnya. Kami sadar ada banyak hal yang masih harus dilakukan untuk maju bersamamu. Karenanya, pelatihan tercipta untuk kami, supaya bisa selalu siaga membantumu. Dan untukmu, kembangkan kemampuan bertani dengan bantuan ilmu serta teknologi terkini. Kami juga selalu siap menjawab setiap pertanyaanmu. Di setiap pendampingan yang kamu perlukan, kami ada untukmu. Dan ketika kami melihatmu berhasil, mendapat kesempatan, serta mencapai apa yang kamu mimpikan, itulah pencapaian terbesar kami dalam menjadi sahabat yang paling baik bagimu. Terima kasih telah berjalan bersama kami selama 30 tahun ini. Mari terus menjadi sahabat dalam berpuluh tahun mendatang. ICFAS Indonesia, sahabat petani yang paling baik.
Indonesia terdapat 14,3 juta hektar lahan perkebunan kelapa sawit. Selama ini lahan perkebunan tersebut hanya dimanfaatkan untuk pertanaman monokrop. Padahal produktivitas dan efisiensi lahan dapat ditingkatkan dengan diversifikasi, salah satunya dengan mengintegrasikan perkebunan sawit dengan pembiakan sapi. Lahan perkebunan yang sangat luas berpotensi sebagai sumber pakan hijauan murah untuk penggembalaan sapi. Dari sisi demand, Indonesia sangat membutuhkan pasokan anak sapi atau sapi bakalan untuk digemukkan. Pemerintah tengah menggalakkan pembiakan sapi di dalam negeri untuk mengurangi beban impor. Beberapa perusahaan telah menerapkan integrasi perkebunan sawit dengan pembiakan sapi ini. Misalnya PT Buana Karya Bakti di Kalimantan Selatan dan PT Kalteng Andini Palma Lestari di Kalimantan Tengah. Usaha yang mereka lakukan dinamakan Sistem Integrasi Sapi Kelapa Sawit, disingkat SISKA.
Manajemen kebun ternyata bisa mengintegrasikan sawit dengan sapi, yang kemudian bisa menghasilkan beberapa hal yang kami anggap itu adalah sesuatu yang baik. Suatu saat ini akan bisa menjadi bisnis alternatif sebagai diversifikasi.
Di lahan kelapa sawit tumbuh berbagai macam rumput yang merupakan gulma untuk perkebunan sawit. Namun, sebagian merupakan rumput yang bisa dimanfaatkan sebagai pakan sapi. Sapi dikembalakan di bawah sawit, dan mereka bisa merumput dengan bebas. Dalam model SISKA, kebun sawit dibagi dalam beberapa blok penggembalaan. Kelompok-kelompok sapi dibiarkan menggembala dan makan di sebuah blok dan pindah ke blok lainnya keesokan harinya. Perputaran ini disebut sebagai rotasi grazing dan akan berjalan selama 3 bulan. Bagi perkebunan, sapi berperan sebagai biological mower yang membersihkan gulma dan mengurangi biaya pembersihan. Perkebunan pun berkurang ketergantungannya terhadap penggunaan herbisida dan bahan kimia lainnya.
Kita dari kebun dituntut untuk bisa efisien mengenai biaya perawatan. Nah, setelah adanya sapi, sekarang kita sangat terbantu, Pak. Pertama, untuk perawatan kita hanya selektif nih. Jadi, untuk gulma-gulma yang terbilang daun lunak, ini tidak kita kendalikan karena nanti bakal jadi makanan sapi. Kita hanya mengendalikan yang sifatnya berbatang keras seperti anakan kayu.
Ini adalah satu bisnis yang sangat efisien. Karena apa? Karena biaya untuk usaha peternakan itu adalah biaya pakan. Nah, ketika pakan itu bisa didapat dari sumber pakan alami yang ada di kebun sawit, yang pasti biayanya akan lebih murah.
Membiarkan sapi di perkebunan sawit tentu bukan hal mudah. Pengelolaan ternak dan perkebunan harus selaras, mengakomodasikan kebutuhan ternak sapi maupun pekerjaan di kebun, dan agar keduanya harmonis. Selain itu, ternak membutuhkan perhatian 24 jam per hari.
Adalah tulang punggung penyedia pangan dan lapangan kerja bagi ratusan juta penduduk Indonesia. Pertanian juga menjadi poros pemeliharaan lingkungan dan pemberdayaan masyarakat pedesaan, memberikan dukungan bagi industri-industri lain dalam meningkatkan perekonomian nasional. Namun, pertanian Indonesia menghadapi berbagai macam tantangan. Perubahan iklim, banjir, kekeringan, alih fungsi lahan pertanian, serangan organisme pengganggu tanaman, penggunaan teknologi 4.0 juga seiring dengan pertumbuhan penduduk dunia yang meningkat pesat, dunia termasuk Indonesia dihadapkan pada ancaman kekurangan pangan. Berbagai cara dilakukan untuk menghadapi tantangan tersebut. Salah satunya, adopsi teknologi pertanian berbasis produk rekayasa genetik dan inovasi pemulihan tanaman. Buah-buahan yang tidak mudah busuk, tahan infeksi tanaman virus, produk tanaman yang sehat dan bebas hama, penggunaan produk perlindungan tanaman yang memenuhi penggunaan teknologi berkelanjutan pada pertanian. Bioteknologi tanaman dalam pertanian selalu memastikan para petani mendapatkan teknologi terbaik untuk kebutuhan pertanian berkelanjutan yang berkontribusi pada ketahanan pangan dengan mengedepankan kajian ilmiah untuk memastikan keamanan pangan, pakan, dan lingkungan. CropLife Indonesia merupakan bagian dari federasi global yang beranggotakan asosiasi regional dan nasional dari 91 negara. CropLife Indonesia merupakan asosiasi nirlaba yang mewakili kepentingan petani industri benih dan pestisida. CropLife Indonesia bertujuan mempromosikan pertanian berkelanjutan untuk kepentingan petani, konsumen, dan keberlanjutan lingkungan hidup. Sebagai organisasi yang mewakili kepentingan petani, konsumen, industri benih dan pestisida, CropLife Indonesia berkomitmen menjunjung tinggi kajian ilmiah dan mendukung pelaksanaan peraturan pemerintah demi terwujudnya penyediaan teknologi baru bagi pertanian. Berfokus pada penelitian, pengembangan, dan edukasi pertanian untuk menghasilkan manfaat maksimal pada penggunaan yang aman dan berkelanjutan pada produk pertanian. Memerangi dan memberantas produk pertanian palsu dan ilegal yang bersinergi dengan pemerintah dan aparat penegak hukum. Meningkatkan pengetahuan dan pemahaman publik pada industri, adopsi teknologi, dan mitigasi risiko pertanian untuk meningkatkan kualitas hasil pertanian. Produksi produk perlindungan tanaman yang aman dan berkualitas, serta penggunaan teknologi berkelanjutan pada pertanian. Divisi pada CropLife Indonesia berbagi peran demi kepentingan petani, industri benih, dan pestisida. Sebagai mitra bagi pemerintah dalam mendukung pelaksanaan peraturan dengan menjunjung tinggi kajian ilmiah yang bersifat objektif dan dapat dipertanggungjawabkan. Sebagai upaya untuk membimbing petani agar dapat menghasilkan manfaat maksimal dari produk perlindungan tanaman. Sebagai peran dalam penegakan hukum dan kampanye anti pemalsuan yang bersinergi dengan pemerintah. Sebagai upaya peningkatan efektivitas dan efisiensi dalam pengendalian resistensi pestisida. Demi terwujudnya pengetahuan dan pemahaman publik mengenai industri pestisida, mitigasi risiko, dan manfaatnya dalam membantu meningkatkan kualitas hasil pertanian. Sebagai upaya membantu petani memenuhi kebutuhan pertanian dengan bioteknologi tanaman terbaik dan berkelanjutan. CropLife Indonesia akan bersinergi dan menjadi salah satu kekuatan sumber daya pembangunan nasional dalam memajukan industri dan penyaluran produk benih dan bioteknologi perlindungan tanaman. Juga berkontribusi untuk meningkatkan ketersediaan pangan dan standar hidup petani kita, serta konsumen dalam semua aspek, baik ekonomi, sosial, dan lingkungan.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology, Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
A sustainable food production for our growing world population requires modern breeding strategies and effective quality control procedures for agriculture and aquaculture. This can be achieved through the production of high-quality seeds and improved yield in plant and seafood farming. An important tool for breeding is the determination of ploidy levels. The ploidy level of an organism is characterized by the number of chromosomes pairs. This is specific to each species. Deviations from regular ploidy levels can have positive or negative effects on the individual. An increased number of chromosome sets, called polyploidy, can produce faster growing and bigger plants or seafood, or create plants with an increased production of secondary metabolites like essential oils. To get plants with seedless fruits, plant breeders produce triploid, sterile individuals. Checking the ploidy levels of such plants guarantees that only seedless fruits are formed. Negative effects can be induced by aneuploidy, characterized by an abnormal number of chromosomes. Aneuploid plants are often stunted. This makes checking for aneuploidy important in quality control of seed production. In all these instances, checking ploidy levels helps to improve yield, quality, and productivity. So farmers gain more benefits with the same amount of investment. For more than 35 years, Sysmex Partech has been the leading supplier of complete solutions for ploidy analysis, offering a broad range of dedicated reagents, instruments, and analyzing software optimized for different sample material and organisms like rice, potatoes, sugarcane, corn, and oysters. Even challenging oxidized plant material becomes easy to handle with our solutions. Sysmex's unique complete solution for ploidy analysis with dedicated instruments, reagents, and analysis software like the automated ploidy detection tool support manual and automated high-throughput measures and analysis in accordance with ISO and REACH guidelines. Start now and benefit from reliable, high-quality and cost-efficient solutions based on the Sysmex experience.
ITS Sains Indonesia merupakan salah satu divisi bisnis dari ITS Group yang berkantor utama di Singapura. ITS Sains Indonesia menawarkan berbagai macam instrumen analitik dan medis, peralatan laboratorium umum, bahan kimia, dan bahan habis pakai lainnya. Selama lebih dari 40 tahun, ITS Group bergerak maju dengan visi memberikan pengaruh di dalam industri yang kami jalani. Kultur perusahaan kami untuk selalu mengedepankan profesionalisme, partnership, dan performance. ITS Group tersebar di berbagai negara Asia Tenggara, yaitu Malaysia, Singapura, Thailand, Vietnam, Indonesia, Filipina, dan Kamboja. ITS Sains Indonesia memiliki 2 kantor yang berlokasi di Kelapa Gading, Jakarta Utara, dan Sukomanunggal, Surabaya. Berikut portfolio produk yang kami representasikan di Indonesia. Produk-produk yang kami representasikan merupakan produk-produk unggulan di bidangnya. Selain itu, kami juga bangga bekerja sama dengan berbagai pihak, mulai dari lembaga pemerintah, industri, hingga universitas. Bila Anda memerlukan bantuan untuk laboratorium Anda, hubungi kami. ITS Sains Indonesia, Bridging Science Inspiring Possibilities.
Wakil Presiden Republik Indonesia tiba di tempat acara.
Besar harapan kami agar fasilitas riset pangan BRIN ini pada akhirnya dapat mendukung pengembangan produk halal dan sehat khas Indonesia, terutama produk-produk yang berasal dari bahan lokal dan komoditas pangan tradisional.
Riset dan inovasi lah yang mendasari pertumbuhan dunia usaha dan juga menciptakan lapangan kerja yang akan mendorong pertumbuhan ekonomi negara.
Kapok Rantani Maju yang posisinya ada di Desa Wonorejo, Kecamatan Satui, Kabupaten Tanah Sumbung. Bagi kelompok tani kita bermanfaat sekali, menekan biaya operasional, baik dari peternak yang biasanya mengembala itu menghabiskan dalam 2 hari itu 3 liter bensin. Manfaatnya itu satu, keduanya dari tingkat keamanan, dari tingkat pemeliharaan kita bisa terkontrol. Mudah-mudahan dengan adanya komunikasi dengan perusahaan kita bisa maju Pak di bidang ternak sapi ini.
Kalau ini kan dijaga baik-baik.
Nama saya Sarip, asisten Abdeling Wonorejo. Dengan adanya sapi di kebun itu, salah satunya adalah mengurangi biaya-biaya operasional yang ada di Abdeling. Kita ada koordinasi mengenai rotasi grazing, kemudian kita berhubungan dengan rotasi panen. Jadi itu yang sebagai patokan kami di Abdeling. Kami pegang ada semacam peta yang dibentuk, yang diolah rotasi dari blok ke blok. Kalau di sini untuk sementara rencananya semua blok Pak. Jadi 1052 hektar.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies. National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by 12 research institutes, have 85 research centers, Executive Secretariat, Executive Inspectorate, Center for Data and Information, Center for Technology Services, and Polytechnic Institute of Nuclear Technology Indonesia. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Excellencies, ladies and gentlemen, welcome back to the main room of the First International Conference on Food and Agricultural Sciences, ICFAS 2002. Next to our agenda, and as a tribute to the participants, is the announcement of the best presenter, the best paper, and the best participants that will be delivered by the Vice of the ICFAS Committee 2022, Bapak Dr. Windu Negara. Before Bapak Windu announce the winner, please kindly for all of the attendees to open the video to help the committee spotlight to the winners. Please welcome Bapak Dr. Windu Negara. The screen is yours.
Thank you, Ms. Anna. Ladies and gentlemen, all the participants, I hope you are still enjoy all the event of the ICFAS, the first international conference of food and agriculture sciences. And then on the behalf of the organizing committee, I would like to express our gratitude and thank you for all of you, all participants, to stay following all the event of the first international of the food and agricultural sciences. And now we are in the most happiest moment for all of us, also for me. I would like to say some people who will reward as the best participant, best presenter, and then the best paper. For the first event, I would like to inform you the best participant from our conference. The first one is Pak Diki Pamungkas. Let's give applause to Pak Diki Pamungkas.
Thank you.
Pak Diki Pamungkas from the Research Center for the Animal Husbandry. So thank you, Pak Diki Pamungkas, and then good luck for the best participant. And then the second one, Pak Diki Pamukas, okay. Good luck, Pak Diki. Congratulations for your reward as the best participant. Don't forget to contact the organizing committee for the reward. And then the second one, Ibu Risa Antari. Congratulations for Ibu Risa Antari. Are you still in here, Bu Risa? Bu Risa also from the Research Center for Animal Husbandry. Okay, now we will go to the next best participant, the last one, Ibu Sri Hartarti from the Research Center for Horticultural and Estate Crops. Congratulations for Ibu Sri Hartarti.
Wah, diborong.
No, this is from the horticultural and estate crops. Yeah, similar, Bu, I know. Bu Sri Hartati, are you still here? From the Research Center for Horticultural and Estate Crops. Okay, congratulations, Bu Sri Hartati. Okay, next one. We are going to the best paper. So, so we already selected 1, 2, 3, 4, 5, 6, 7, 8. 8 the best paper based on the paper categories. The first one for the category the appropriate technology, and the winner is let me see. Okay, Pak Astu Unadi with the title Design and Development of Semi-Automatic Grafting Machine for Cocoa. And then Pak Astu Unadi is for the from the Research Center for Appropriate Technology.
Thank you.
Congratulations, Pak Astu.
Thank you.
Now we are going to the next category, Food and Technology Processing. And the winner is, I don't know, this is Bu or Pak Radestia Triwibowo from the Research Center for Food and Technology Processing with the title Occurrence and Identification of Zoonotic Anisakis sp from frozen imported fish in Indonesia. Pak Radestia Triwibowo, congratulations.
Thank you for moderator.
Okay, thank you. The next one for the category agro industry, and the winner is Ferry Manoy.
Ya, Pak Sarep.
From the Research Center for Agro Industry. The title of the paper is the nutmeg seed storage technology using various packaging and temperature for aflatoxin reduction. Congratulations, Bu Ferry Manoy. Okay, we go to the next category, livestock production. And the best paper for the livestock production category is Slamet Diah Volkandari from the Research Center for Food and Technology Processing with the paper title, Phylogenetic Relationship for Kisar Sheep from Kisar Island, Southwest Maluku based on MT-ND1 gene. Bu Slamet Diah Volkandari, are you in here? Okay. Congratulations, Pak Munir. Don't worry, ladies and gentlemen, we still have 1, 2, 3, 4 more categories. The next one, plant breeding and biotechnology. The winner is Kusumawati Kusumanegara from a Research Center for Horticultural and Estate Crops with the title Molecular Analysis and Evaluation on Fruit Performance of Genetically Engineered Tomato Lines with Double Virus Resistance. Ibu Kusumawati, congratulations for your achievement.
Thank you.
Can you show the video so we can take a picture?
Thank you.
And then all right, next we'll go to the agronomy category. And then based on our review, the best paper for this category is belong to Matias Pratama from the research center. Oh, the Bureau of Organization and Human Resources. Pak Matias Pratama, are you with us? With the title of the paper, Maintaining Soil Properties and Increasing Cabbage Yields Through Minimal Tillage and Legume Cover Crops. Congratulations for Pak Matias, or maybe there is a team of the Matias that can show in this conference. Rini Rosliani, Trihandayani, Prama Yufdi, Ine Sulastrini, and Darkam Musadat. Ibu Ine, Ibu Trihandayani. So are you the team of this article?
Yes, sir. Thank you very much.
Okay, congratulations Bu Ine, Bu Tri. Congratulations for your achievement. Okay, next we will go to the plant protection. And then the best paper for this category is belong to S. Hardianti from the Research Center for Horticulture and Estate Crops. The title for the paper, hello. Hello, Bu. Hello. Congratulations, Ibu. You are very busy, Bu. I know. The best researcher always busy. The title for her paper is Maintaining Soil Property. Trapped volatile organic compounds produced by bacterial antagonists affect the growth of Sclerotium ropsi. Again, congratulations, Bu Siti Hardianti and the team.
Thank you.
You're welcome. Okay, the last one, the last category, soil science.
Tadi lagi dipanggil.
Ya, Bu Sri, selamat. Okay, the next one for soil science category. The winner is Mas Teddy Sutriadi from Research Center for Food Crops. The title, congratulations, Pak Mas Teddy. The title
Thank you very
is,
much.
Okay, congratulations. Dynamic of soil phosphorus fraction and its correlation with maize yield on organic acid application with phosphate rock in upland acid soil. Okay, so this is the last best paper for soil science category. Congratulations, Pak Mas Teddy. And then the last one, the last event is category is about the best presenter. So please prepare yourself, ladies and gentlemen, all the presenters in this conference. We also have the winner based on the category. The first one, the appropriate technology. The winner is Diang Sagita from the Research Center for Appropriate Technology. Big applause for Diang Sagita. Hello, Bu Diang, are you in here? Oh, Pak, I'm sorry, Pak. My apology, Pak Diang. Pak Diang? Okay, not in here. We go to the next category. Food and Technology Processing. The winner is Ibu Rianti Eka Fitri from the Research Center for Appropriate Technology. Congratulations, Bu Rianti. And then next, the category of agro-industry. The winner is Agus Eko Cahyono, the Research Center for Agro-Industry. Congratulations, Pak Agus. Okay, next, for the livestock production. This name is familiar for me because this is one of the moderator in the livestock production. The winner is Ibu Maureen Krisya Hadiarti from Research Center for Animal Husbandry. Congratulations, Ibu. Big applause for Ibu. Okay, next one. It's okay, Ibu. This is surprise. So for the presenter, please prepare. Our team will spotlight your video. Next, the plant breeding and biotechnology. The winner is Kristianto Nugroho from the Research Center for Horticultural and Steppe Crop. Pak Kristianto, can you find? No? Okay, thank you. The next one, agronomy. The winner is Ibu Diah Setiorini from Research Center for Food Crops. Congratulations, Ibu Diah. Okay, next, plant protection. The winner is I don't know, this is either Ibu or Bapak Cairani. Ibu, congratulations, Ibu.
Thank you.
Ibu
Thank
Dia,
you. It's a surprise for me.
Ibu Cairani,
Hey,
nice to meet you, Ibu. Congratulations.
Thank you.
Ibu Cairani is from the Research Center for Food Crops also.
Yes, that's correct.
Thank you. And then the last winner from the soil science category, the best presenter for this category, Ibu Gagat Restu Pratiwi from Research Center for Food Crops. Congratulations for Ibu Gagat. Ibu Gagat not in here? No. Okay. Ladies and gentlemen, for the winner, congratulations for all the winner. And then I will return the time to the MC, Miss Anna. Thank you very much.
Thank you very much, Pak Windu, for announcing the winner. And once again, big congratulations to all the winner. May this accomplishment bring you more enthusiasm to continue the journey in research and innovation. So, ladies and gentlemen, move to our last agenda. It is our honor to have the chairman of the Research Organization for Agriculture and Food to deliver a speech, and afterward, we would like to request the chairman to officially close the conference. Please welcome Ibu Puji Lestari, PhD. The floor is yours.
Thank you, Anna. Assalamualaikum warahmatullahi wabarakatuh. A very good afternoon, ladies and gentlemen. I hope most of participants are still joining this the end of this session. So, on behalf of Research Organization for Agriculture and Food BRIN and the organizing committee, I am very delighted to deliver this closing remark on the success of this first ICFAS 2022, which has been truly a great 2 days. I believe that during these 2 days, we all had insightful, interactive discussion and a great chance to share the outcomes of our research. It is first ICFAS with the main theme, advanced agricultural technology to deal with climate change issues for achieving food security. Held virtually, we have learned a lot from the distinguished keynote speakers and plenary sessions, which were delivered by 10 invited speakers who have shared their remarkable works and experiences. I would also like to thank our presenters and participants who have joined this conference from various countries. More than 300 interesting scientific papers were presented in the parallel session, and I am confident that every presenter delivered valuable knowledge in their respective fields. On behalf of the committee, thank you all for delivering such important material to broaden and deepen our insight and knowledge. I sincerely hope that through your presentation and discussion afterward, we can all contribute to address the agricultural and food issues in our own countries. My appreciation is also addressed to all the moderators of all sessions for their charming leads in bringing this session to fruitful discussion. Furthermore, I convey my congratulations to the best presenters, the best papers, and the best participants. Your contribution and participation in our event are inspiring. And at this moment, also, I would like to express my impression. That's very awesome that congratulations to organizing committee and team and all of us, I think, in this ICFAS, that on the fantastic number of viewers of ICFAS in live streaming YouTube, reaching, I don't know, around maybe more than 2,000 viewers on day one. I don't know, day two. That is very amazing. Thank you so much for all. And ladies and gentlemen, I wish you have all enjoyed networking opportunities throughout the conference. I am truly satisfied that our guests and speakers were able to inspire each other on advanced agriculture and food technologies. We had enlightenment through the discussion of recent agricultural technology to respond to global climate change, the utilization of artificial intelligence and blockchain technology in the circular economy to achieve a sustainable food supply chain, cattle oil palm integration system, marine resources for future food development, technologies to reduce greenhouse gas emission from various agribusiness, metabolomics, and other technologies to improve food quality and reduce food loss. And with the latest biotechnology development, including regulation and commercialization aspects and other related topics in this event. I hope all of us can take advantage of our networking and knowledge sharing at this conference to improve our best practices in agriculture and food technology, especially to overcome climate change impact and achieve food security. Ladies and gentlemen, we at the Research Organization for Agriculture and Food, BRIN, are pleasure to be of service to all fellow researchers and collaborators by thus providing platform for establishing a dialogue and expert networks that we are convinced to be useful for all of us. But even more than that, we are excited to see practical complication and concrete steps made at different levels. It requires, of course, further hard work as well as intellectual aspect, financial and human resources to fulfill this intention. With the support of the grassroots to the policy-making level, we hope changes are being planned as we speak now and significant improvement on research and innovation to deal with climate change issue for achieving the security that will show impact in the near future. Excellencies, distinguished guests, ladies and gentlemen, on behalf of BRIN, our agency, we humbly apologize for any deficiencies and shortcomings during the implementation of this event. In the end, to please all join me to give big hands to all members of the scientific committee and staff from the organizing committee and all parties who have contributed their best effort, hard working, and assistance in organizing the event and ensuring the success of it. I do hope that the first ICFAS in 2022 is able to give impact all of us to greatly foster and empower the society through agricultural and food science and technology for sustainable innovation. To conclude, I think I have to obviously closing, right? So okay, by the blessing of Allah, Alhamdulillah, I officially close this first ICFAS in 2022. And to take applause for all of us. And then to conclude, thank you for the great contribution and looking forward to seeing you in the next year ICFAS. May our protect and bless us. Have a nice weekend all. Thank you so much for this successful event. I'm very happy to know the journey. Okay, I return to the master ceremony. Thank you very much. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Alhamdulillah. What an enlightening speech. Thank you very much.
Congratulations.
Congratulations to all.
Congratulations for
Selamat untuk panitia yang sudah bekerja keras.
Excuse me, excellencies, ladies and gentlemen.
Terima kasih panitia.
Okay, what an enlightening speech. Thank you very much, Ibu Puji Lestari, PhD, the chairman of the Research Organization for Agriculture and Food. Excellencies, ladies and gentlemen, finally, we come to an end on the first International Conference on Food and Agricultural Sciences, ICFAS 2022, with the theme Advanced Agricultural Technology to Deal with Climate Change Issue for Achieving Food Security. We would like to thank you for spending your time with us during these 2 days. Thank you for the active participation. Thank you for our speakers, moderator, and the organizing committee. We also want to express our deepest gratitude to our sponsor for supporting our conference, Indonesian Association of Integrated Cattle Oil Palm Entrepreneurs and Observers, or Gabungan Pelaku dan Pemerhati Integrasi Sapi Sawit Gapensiska Indonesia, Indonesian Biotechnology Consortium, CropLife Aid Indonesia, ITS Science Indonesia, ICFAS Indonesia, Elokarsa Utama, Cultiva Life Sciences, Indonesian Breeding Science Society or Perhimpunan Ilmu Pemuliaan Indonesia or PERIPI, Indonesian Agronomy Society or Perhimpunan Agronomi Indonesia or PERAGI, and the last but not least to all distinguished participants. We would like to remind you that even though we are getting recovered from the pandemic of COVID-19, please keep practice the health protocols properly, keep the physical distancing, wash your hand regularly, and please wearing a mask everywhere you go. Let's take care of each other and stay safe and healthy. And finally, on behalf of the organizing committee, we would like to apologize for the inconvenience matters during the program. Thank you and see you at ICFAS 2023. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Terima kasih, Mbak Ana.
Terima kasih semuanya. Terima
Teman-teman
kasih
semua.
panitia.
Panitia sukses.
Selamat Bu Puji.
Terima kasih untuk panitia.
Kita kan nanti ketemu di situ kan? Nah,
Peserta semuanya presenter banyak kantor kegiatan
Ya.
Mohon izin Lip, Bapak Ibu semua.
Izin lift ya.
Sampai
ketemu lagi.
Alhamdulillah ya Bu.
Ya, senang ya Allah. Tuh, Pak Mulyana.
Alhamdulillah semuanya.
Pak Mulyana baru nongol.
Ada dari tadi.
Oh, dari tadi.
Ada Pak. Ada Pak.
Oke, Pak Mulyana. Ini masih di Serpong kah?
Enggak apa di jalan biasa.
Oh,
ini Pak Mul di Kerawang ya, Pak Mul ya, di Kerawang.
Oh iya, temannya Pak Maijan. Pak Maijan, apa kabar Pak Maijan?
Kabar baik Pak Tri. Ini di Kusnoto.
Oh masih di Kusnoto. Oh, sama. Saya juga di Kusnoto nih Pak Maijan. Saya di lantai 5.
Oh iya. Tadi juga ketemu Ibu Ana ya.
Ana ya. Oh iya.
Ya Pak Patri selamat ya. Alhamdulillahirabbil alamin.
Iya, terima kasih.
Alhamdulillah. Lancar semuanya, alhamdulillah.
Semuanya.
Bu Puji ya, alhamdulillah Bu.
Terima kasih.
Sukses untuk kita semua.
Ya, untuk kita semua jelas ya, alhamdulillahirobbilalamin.
Izin lift.
Izin nih Bapak Ibu.
Iya Pak.
Selamat ya Pak Tri.
Oh ya baik Pak Cepi, makasih.
Ya sama-sama Pak Mulyana, sukses ya.
Ya sama-sama Pak.
Insya Allah tahun depan dilanjutkan Pak Tri ya.
Iya, ya, Insya Allah Pak. Ya sampai ketemu ya Pak.
Ya, Insya Allah.
Ya, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi. Pak Arif, apa kabar?